Good morning, one and all. I, Ms. Muskan Sharma, on behalf of School of Science, Sandip University, Nashik, and Society for Green Environment, New Delhi, India, is pleased to welcome you all back on the third day session of e-conference on current trends in waste treatment, reuse, and valorization. We are going to start shortly. Good morning, one and all. I feel happy to invite today's first expert, Professor Vinod Kumar Garg, Doctor and Professor Dean, Department of Environmental Science and Technology at Central University of Punjab, Bhatinda, India, and Dr. Ravi Kiran Pagare, Associate Professor, Sandip University, Nashik, to chair the session. I now request Dr. Pagare to please introduce our expert.
Thank you, Muskan Madam. Uh, I welcome you all on behalf of School of Science, Sandip University. Uh, I welcome in international conference on current trends in waste treatment, reuse, and filtration. Okay. Today, first session, Professor Vinod Garg is there with us. Professor Vinod Garg, he is working as a professor in Department of Environmental Science and Technology, Central University of Punjab, Bhatinda. Previously, he was worked as a Guru Jamshedwar University of Science and Technology, Haryana. Professor Garg is well-rounded research with more than 30 years experience in leading, supervising, and undertaking research in the broad field of solid and hazardous waste management. He had his research group and working on water and wastewater pollution monitoring, solid waste management, pesticide degradation. He has published more than 200 research papers, reviews, articles, and 22 proceedings, and 6 editorial in peer-reviewed journal. His citation is 15,000 and H index 65. So it tells that sir is having vast experience in his field. He has published two books and 12 book chapters. He has completed 10 sponsored research projects from various agencies. The important thing, he has awarded with the Thomson Return Research Excellence India Citation Award 2012. He has guided 24 PhD students and 80 MSc students for the research work. He is an active member of various scientific societies, mainly in Indian Science Congress, Biotech Research Society of India, National Solid Waste Association of India, Indian Nuclear Society. He was elected a fellow of Biotech Research Society of India, and he has served on editorial board of various scientific journals in his research area. So thank you, sir, for accepting our invitation as a plenary speaker in our international conference. Your topic, that is nutrient recovery from solid waste using vermi technology, really it will be very helpful for the scientific society and also for the social benefits also. Sir, I welcome you for your speech. Thank you very much, sir. Over to you,
Thank
sir.
you, Dr. Ravi Kiran, for this wonderful introduction. I need just one confirmation whether I'm audible or not. Can anybody confirm me whether I'm audible or not? Okay. I'm thankful to Sandip University that they have given me a chance to share my views on the solid waste management. Friends, you know, on present day, we are facing a number of environmental problems, and solid waste is one among the major environmental problem which we are facing at present. Solid waste are generated, I think, by every activity of the man. Presently, you can say number of methods are used by people for the management of solid waste, but most of the methods which are used presently, they are destructive in nature. So we have to look for such methods which are not destructive in nature and which can be used to recover different kind of nutrients which are present in the solid waste. Because if we use destructive methods, those nutrients which are present in the solid waste can be destroyed forever. So I'm going to share with you my presentation on this topic. Uh, so let me share my screen with you. I don't know how. Yes. So friends, today we will be discussing that how this vermi technology, that is a biological method, can be used for the management of solid waste. Friends, before the management of solid waste or before applying any technology for the management of solid waste, we have to understand the nature of the solid waste. Only then we can choose, we can select a technology. As far as solid waste are concerned, they are highly heterogeneous in nature. And because of that heterogeneity in their nature, no single method is sufficient for the treatment or for the management of solid waste. You know, the reason of heterogeneity is that solid waste are generated from a number of sectors like domestic sector, agricultural sector, industrial sector, commercial sector, etc. At present, about 0.2 to 0.6 kilogram of solid waste per capita per day is produced in India. Recently, I was reading one report that if all the solid waste which is generated in India is loaded in the trucks, and if we make a line of head to tail of those trucks, the line will be 300-kilometer long from Delhi to Samagra or Mathura, something like that. This much solid waste is generated in India. As far as waste collection efficiency of India is concerned, in bigger cities, bigger size cities, the waste collection efficiency is 70 to 90%, whereas in smaller cities, it is 50 to 60%. Friends, you know that waste is collected, but hardly 5% of the total budget of the urban local bodies is spent on the treatment of the solid waste. Most of the cost of the this budget of the urban local bodies is going just for the collection and the transportation of the waste. Friends, these are some of the photographs which I have clicked while interacting with the rag pickers. And this is a very, you can say, common scene in the developing countries, particularly in India. While interacting with these people, I found that these people are getting their livelihood by finding something that is present in the waste. So based upon these photographs, we can very safely presume that these wastes are containing something, they are containing some resource, and that resource is the reason of their livelihood. You can see the animals are trying to find out something, these rag pickers are recovering something. So at least we can say one thing: the solid waste, which we say waste, is really is not a waste. That is containing something that is valuable, and we should recover that valuable material from that. Friends, you know how difficult this is nowadays to acquire the land for the disposal of solid waste, or rather leave solid for solid waste. For any other purpose, it's very difficult to acquire the land. Here, based upon a research paper, I have given the projections that how much land will be required after 100 years of independence in 2047. So that time we will be requiring about 1,300 square kilometer of the land in our country just for the disposal of solid waste. Friends, we know that we are a highly populated country and we cannot spare this much land for the disposal of solid waste. We have to look for certain methods by which we can recover the valuable things which are present in the waste. If you look for the composition of the Indian solid waste, you will find it contains 40 to 50% of the biological, biodegradable, non-toxic waste that can be used or that can be recovered making the use of some biological method, and one such biological method is vermicomposting. Friends, here I have shown different kind of methods which are presently used for the disposal of solid waste. Open dumping and open burning, these are the two very commonly used methods, practices, but these are the most dangerous methods for the, you can say, environment as well as for the human beings. Hog feeding of the waste to the pigs is also a common sight in India. But you know that this feeding of hogs, pigs, on the raw waste can lead to several problem in those people those eat pork. Lead filling, yes, this is considered as one of the ultimate method for the disposal of solid waste, but this method require land, and I have already told that we are very short of land. Incineration, yes, this is also called as ultimate method of the disposal of solid waste. But in this method, we have to burn the temperature at a temperature of 800 to 1100 degrees Celsius. And for that burning, we require energy. Recently, a study was carried out, and according to that study, most of the incinerators in India are operating at a temperature of 400 to 500 degrees Celsius. When these incinerators operate at a lower temperature, they lead to production of many toxic gases like dioxins and furans, which are carcinogenic. Recycling, a very good approach. Different kind of waste which are non-toxic can be recycled. Pyrolysis, biomethanation, these are the energy recovery methods. Then agricultural waste, they can be used as livestock. Similarly, some agricultural waste can be converted into briquettes. And these are the two biological methods where the non-toxic biodegradable waste, making the use of composting and vermicomposting, can be converted into manure, and that manure can be used in our agricultural fields. Friends, you all might be knowing that agricultural fields in India are deficit in organic matter. Reason being, in Indian soils, because of the temperature which we have in our country, the microbial activity in the Indian soils is quite high. And secondly, chemical reactions also take place at a very fast rate. That's why the organic matter of Indian soils is depleted very fast, and we have to add this organic matter and again to maintain the fertility of the soil in India. So today I'll be talking to you about this one method, that is vermi technology, that how vermi technology can be used to manage the solid waste. Friends, in case of vermi technology or vermi composting, we make the use of earthworms, and these earthworms convert the non-toxic biodegradable waste into a manure that is called as vermicompost, that is having very good manurial values. Friends, there are more than 4,000 species of the earthworms on this earth. All kind of earthworms cannot be used for the purpose of vermicomposting. Depending upon the niche, depending upon the fact that where the earthworms live in the earth, these can be divided into three ecological groups. These are epigeic earthworms, endogeic earthworms, and anasic earthworms. Epigeic earthworms are free living. These live upon the earth. These are solitary in nature. These can move from one place to the other place in search of food. Endogeic, as the name indicating, these are living inside the earth. And anasic earthworms, these live outside the earth. Among these all three groups, epigeic earthworms are known and well tested for their potential for the vermicomposting. So here I have shown, as I told you, that all earthworms cannot be used, all species cannot be used for the purpose of vermicomposting. There are certain species, they should have certain characteristics. So what are those characteristics? The earthworm species which are suitable for vermicomposting, they should have wide adaptability to the environmental factors. If you come to northern India, here you will find in winters temperature may go up to 2 degrees Celsius, and in summer temperature may go up to 40 degrees Celsius. So the earthworm which you use for the purpose of vermicomposting, that should be able to tolerate that temperature, as well as the earthworm species which we use or which you use for the purpose of vermicomposting, that should be adaptable to different kind of organic material, organic waste, because as I told you, the waste is highly heterogeneous in nature. The earthworms which we use, they should have very high growth and reproduction rate. They should have very high feeding rate. If they have high feeding rate, only then they can convert large quantities of the waste into vermicompost. And the species which we use for the purpose of vermicomposting, that should have lesser inactivity period to the new feeds, because whenever a different kind of waste is given, earthworms may take a lot of time to adapt to that. So that species which we use, that should take minimum time to adapt to the new feed. Nowadays people are working using the polyculture for the purpose of vermicomposting. Polyculture means different species of the earthworms are added in the same vermi bin. So the earthworms which you use, the species of earthworm which you use, that should be compatible, or that should be tolerant to other species which are present in the vermicomposting bin. Friends, I told you earlier that more than 4,000 species of the earthworms are present on this earth. About 520 species of the earthworms are found in India. Among them, just 20 species are good for the purpose of vermicomposting. And among them, these are most widely used species of the earthworms. Icenia fetida is one such earthworm species that is most widely used for the purpose of vermicomposting. This also called as red vigilers. Friend, this is not an Indian species. This has been imported. Then we have Icenia underi. This species is widely used in southern part of the country. And then Idrillus, Eugenie, Perionox, Lumbericus, and Lampito. These are some other worms. But among these all, Icenia fetida is the most widely species that is used for the purpose of vermicomposting. Friends, when we do vermicomposting, this is not only the earthworms which play the major role. Along with earthworms, many other things like mites, flies, predatory planarians, centipede, millipede, springtails, bacteria, fungi, etc. are present in the vermicomposting bin, in that bin in which you are doing the vermicomposting. So many things are present. Actually, the role of earthworm is there to make the environment, to make the conditions very conducive to other species which promote the process of vermicomposting. So with this background, a lot of work is going on in our lab on vermicomposting of different kind of sludges, different kind of waste. Some of the experiments which we are doing, I'm going to share with you. Friends, this is one project that was sanctioned to us by the government of Haryana, that we all know that a lot of sludge is produced in the sewage treatment plant in STPs. And you might have seen many a times that sewage sludge, dried sludge, is not used for any purpose, that is disposed along the road. Sometime it is disposed in the agricultural form, in raw form in the agricultural fields, where it may be problematic. So we were sanctioned one project to find out that what to do of this sludge. And we recommended the government of Haryana that we can do the vermicomposting of this sludge, and after vermicomposting, this sludge can be used as a manure in agricultural fields. So friends, for that we collected sewage sludge from a STP that is located in the village Dhanwapur near Gurgaon. Gurgaon has been declared by the government of Haryana as green city. And the capacity of this plant is 15 MLD. That is a very big plant. The sludge which we collected, at the time of collection, it was foul smelling, and earthworms are very sensitive to the smell. So we dried it in the sunlight. So that smell goes away. And for a week about, we dried that, and we did periodic turnings also. Then we screened this sludge to remove all non biodegradable material like brick pieces, some stone, glass pieces, metal pieces, and every non biodegradable material was removed from it. Friends, this sewage sludge is not the primary food of the earthworms. If we add earthworms in this sewage sludge, as such, earthworms will die within few hours. So for that, we have to add some other bulking agent, some other material into this sludge to make it palatable, to make it eatable to the earthworms. The other material which we used, that was the cow dung. And then we mix the cow dung in different ratios uh, in this sludge, and then we did its vermicomposting. So we did two kind of experiment. First one was, what is the appropriate ratio of the cow dung and sewage sludge that is acceptable to the earthworms for the purpose of vermicomposting. Secondly, what is the growth and reproduction of the earthworm in different mixtures of this sewage sludge and cow dung. So whenever we do this kind of experiments, friends, we do study of the two kinds of parameters. One are the biological parameters which are related with the earthworms, and second are the physicochemical studies of the vermicompost. So the very first thing which we did, we analyzed this cow dung and the sludge for different parameters. And nowadays vermicompost has been added in the Fertilizer Act of India, and there certain limits for different parameters are given, including the heavy metals also, that the heavy metal load of the vermicompost should not be more than this, like that. So these all parameters like pH, electrical conductivity, ash content, nitrogen, phosphorus, potassium, CN ratio, and different kind of trace metals and heavy metals were analyzed. And we found that chromium was a little bit higher and lead was also a little bit higher. The reason being, maybe some of the industries which are using these metals may be disposing their wastewater in this sewage water. And then we made different kind of vermi bins. We made a total of the 6 vermi bins, which were containing cow dung and sewage sludge in different ratios, like 100% of cow dung, that was our control, then 90% cow dung, 10% of sludge, 80% cow dung, 20% sludge, 70% cow dung, 30% sludge, 60% cow dung, 40% sludge, 50% cow dung, and like that. We did these experiments. We mixed up to 90% of the sludge, but we found that when sludge content was more than 50%, earthworm could not survive into that. So those experiments were terminated, and we continued these 6 feed with these 6 feed mixtures. So friends, these two things were mixed, and these were allowed for pre-composting before adding the earthworms. This is one thing very important. If you mix them and immediately add the earthworms will die because of the smell and because of the heat that is generated. So for approximately 3 weeks, the mixture of cow dung and sludge was allowed to compost. And when it was semi-composted, only then earthworms were introduced into the vermi bins. And then this experiment was continued for 15 weeks. After 15 weeks, we analyzed the characteristics. We analyzed the characteristics of this feed on the 0 day, and then we analyzed on the 21 day when we introduced the earthworms, and then at the time of termination. So I'm going to show some of the results to you of this experiment. So friends, the upper table is showing the characteristics of these 6 feed mixtures. Here you will see, and the lower table is showing the characteristics of the vermicompost that was produced. You will find, friends, that pH was reduced after vermicomposting, and this reduction of pH may be that some acids may be produced by the degradation of different materials. Electrical conductivity has increased during the vermicomposting, and this may be due to the release of certain ions which contribute to the electrical conductivity. Then you will find the ash content has increased significantly than the initial raw materials. Then you will find approximately 2.5 times increase was there in the ash content, that clearly indicate that the mineralization is taking place. Then if you come to the organic carbon content has decreased. Organic carbon content has decreased because carbon has been used by the earthworms, and carbon dioxide is released by the earthworms, and this also indicate the mineralization. Then nitrogen, which is an important nutrient for the growth of plants, that has increased after the vermicomposting. Nitrogen content has increased, you can say, 2 times or some 3 times after vermicomposting. Similarly, potassium content has increased. Friends, here I want to say one thing, that this potassium content has increased due to the reduction in the volume of the waste. If you take 100 kilogram of the waste, at the end of the day you will get about 40 to 50 kilogram of the vermicompost, and this nitrogen has been added because of the microbial activities. So remember this potassium has not been produced by the earthworm. This increase is just due to the reduction in volume. Similarly, phosphorus content has increased, and this increase in phosphorus content is also due to the reduction in the volume of the waste. Friends, CN ratio is an important indicator which indicate whether the waste is stabilized or not. Whenever CN ratio is less than 20, we say that waste is stabilized. So we monitored this at different days, and you will find that with the time, if we move from left to right in every column, that this CN ratio reduced with the time. This reduction in CN ratio is due to two factors which I have told just now. One is reduction in the carbon content and increase in the nitrogen content. And at the end of the day, when experiment was terminated, this CN ratio of the vermicompost was somewhere 10 to 15. That is acceptable CN ratio. And based upon this, we can say that whatever vermicompost we got from the waste, that was highly stabilized. Friend, I told you in the beginning that nowadays this vermicompost has been included in the Fertilizer Act of India, and certain recommendations have been given that what should be the heavy metal content in the vermicompost. So we monitored the heavy metal content in the vermicompost in the initial raw material as well as in the final vermicompost. And based upon this, we found that heavy metal content was within the permissible limits in all the vermicompost which we produced from the mixture of cow dung and sewage sludge generated at the wastewater treatment plant. Friends, we were interested to know the growth of earthworms also. So we, on the weekly basis, we monitored you can say, find out the biomass of the earthworms. For this purpose, we took the earthworms out from every bin on weekly basis. We measured their weight, and they were again put up back in the experiment. So here you will find that initially up to, you can say, 7 weeks, there is increase in the biomass or the weight of the earthworms. After that, it is stabilized, and then this weight of the earthworms started decreasing. Friends, this clearly indicate as long as the biomass of the earthworms is increasing, it means the feed is available to the earthworm. And when it is decreasing, it means all the waste, all the feed has been consumed by the earthworms, and now there is no more feed that is available for the earthworms. So friends, then we monitored different parameters related to the earthworms, and here in this table you will find that the biomass of the earthworms was increased more than 10 times, and maximum biomass was attained in the 6th week as shown in this slide. After that, it started reducing, as I told you earlier. And the growth rate means how much weight was gained by each earthworm. This is average. That was about 17 to about 22 milligram per day in different wormy beans. And here we try to calculate that how much biomass has been gained by the earthworm by per gram of the waste. So about, you can say, 20 to 30 milligram biomass is gained by the earthworm by consuming the 1 gram of the waste. So friends, based upon this experiment, we concluded that if 30 to 40% of the sewage sludge is mixed in the cow dung, it can be vermicomposted, and we get a very good quality vermicompost that can be used in our agricultural fields. And means this material which we get, that is vermicompost, that can be a very good quality manure. Then friends, we did another project that we did with the textile industry, because that the textile industry was having a ETP, effluent treatment plant. Lot of sludge was generated. They were finding it difficult what to do. Then they contacted us that what to do of this sludge, and then we suggested them. Some experiments were conducted by us, and we suggested them that if you mix up to 30% of your sludge in the cow dung, that can be converted into vermicompost, and that vermicompost can be used in agricultural fields. We did experiment with cow dung, and we did experiments while using the biogas plant slurry, and then we did experiment with this poultry droppings, because we have a number of hatcheries here which are producing this chicken. They produce a lot of excreta of the birds. So we try to mix different kind of waste with this sludge of the textile mill, and we were able to convert that into vermicompost. Remember friends, whenever this textile mill sludge was mixed more than 30%, at that time it affected the growth of earthworms. Same thing I have shown here, uh, which is showing the growth of the earthworms. Growth of the earthworms was affected when we mixed more than 30% of this sludge in the cow dung. Similar kind of work we have done for one food industry. You might be knowing Parle-G. Parle-G is also having a ETP, effluent treatment plant, and that is in Bahadurgarh city, that is near to Delhi. And we found that if 40 to 50% of their waste STP, this ETP sludge, is mixed with the cow dung, or you can say this biogas plant slurry, etc., that can also be vermicomposted. Friends, similarly, we did one experiment that whether this domestic waste can be converted into vermicompost or not. So we did a lot of experiments, and based upon that, uh, we concluded that yes, if we mix if we mix this domestic waste with cow dung, that time it can be converted into good quality manure. Here I have shown some of the results, that if we mix cow dung and domestic waste, that time organic matter will decrease with the time. And this indicate again same thing, that mineralization is taking place. Here I have shown that how phosphorus content has increased with the time when cow dung and household waste was mixed, and here I'm showing that how the potassium content has increased. Then same study we did for the heavy metals. Heavy metals again increased in the vermicompost, and this increase in heavy metals of the vermicompost after vermicomposting is again due to reduction in the volume. Otherwise there is not any other factor that is responsible for this. Friends, similarly we did some experiment on the water hyacinth. If you come to the northern part of India, you will see almost all the water bodies have been infested by this weed. This is one among the 10 most troublesome weeds at present which we have Similar situation maybe in southern part of India or western part, I really don't know. So we try to manage this with the help of vermicomposting, because presently what people are doing, they collect it from their village ponds and then throw it outside the pond, whereas this is a very good resource because it is a biological material and it can be a very good quality manure. So we did its vermicomposting and we found that if 25% of water hyacinth is mixed with the cow dung, there is no effect on the quality of this uh, vermicompost at all, and this can be used as a very good quality manure in the agricultural fields. Friends, this is a, you can say, very big accumulator of heavy metals. This plant is a very big accumulator. It's a well-known hyper accumulator of heavy metals. So we did study on the heavy metals also. So whenever you work this kind of weed, always take the heavy metals into your consideration. Although we found that heavy metals were within the permissible limits when we worked on this weed. Friends, this is a recently published paper. Here in northern part of India, rice straw, this stubble, is one of the biggest problem. In the month of December, we get a lot of air pollution, and that is because of the burning of this rice straw. So we did some work on that, whether that rice straw can be managed with the help of vermicomposting or not, and we found very good results that rice straw, which is otherwise burnt by the farmers, can be converted into a very good quality manure with the help of earthworms or while using the vermi technology or vermicomposting. So, friends, beyond these lab experiments, we are trying to develop some potting media also. Nowadays, people are doing, you can say, a lot of gardening activity in urban areas. We are trying to develop some potting media, and we are training the farmers in our area that you take your waste, do its vermicomposting, add some nutrients into that, which we suggest them. So we are doing experiments on that. Some of the photographs I have shown you. Then, friends, the area in which we are living, we are having 5 thermal power plants. A lot of fly ash is generated. So we were interested that what to do of that fly ash. So we are trying to develop certain mixtures of the vermicompost and fly ash that those can be used in agricultural fields, because fly ash also contain many macro and micro elements, nutrients. So we prepared different mixtures and we are testing those mixtures in our agricultural plots. So these are some of the photographs which I am sharing with you. This is my student PDF who is doing these experiments. So here we are mixing this fly ash and vermicompost, and then we are using this mixture in different crops. Here I have shown you some photographs of the mustard crop, and we have found very good results uh, when we use this mixture in the agricultural field. Similarly, we are trying to develop a, you can say, a media for the purpose of mushroom cultivation. Here we are taking a boiler ash from industry, and we are mixing this vermicompost into this, and we are trying to get this mushroom, and very good quality mushroom is grown into that mixture, and we are giving some training to the farmers in that regard also. Yes, then we are interested because in the urban areas, okay, it's okay for the rural areas, but in urban areas there is a shortage of space, shortage of land in the houses. We are trying to develop certain you can say vermi bins, which can be used at the domestic level in the urban areas, and we have made certain design and some results we have published also in different journals. Friends, few more photographs I'll be showing you, then I'll be closing my presentation. Many a times people do vermicomposting in the open. Friends, we don't recommend this kind of vermicomposting because there are a number of pests like birds, rats, which can take away your earthworms. And once earthworms are gone from your bin, I think nothing will be left over there. Then, friends, for urban areas, we are recommending this kind of four chambered bin, because I told you in the beginning, the earthworm species Icenia fetida, that is free living, can move in search of food from one place to the other place. So what we are suggesting, people or different housing societies make this kind of vermi bins. Take your waste for 20-30 days here, that will be composted. In the meantime, you start putting your waste in the second bin. After 30 days, when it is semi composted, add earthworms. When it will be consumed by the earthworms, waste will be consumed, it will be converted into vermicompost, and then earthworms will move through these holes which we have made in this wall in the next chamber, then next to next, then next to next. So this way you can have a cycle of 120 days for the management of your waste. Then, friends, we have developed one more model for the rural areas. We have made certain vermi bins, and in these vermi beans, either we have to make the shed. Here we have recommended the farmers, you grow the jatropha. So here you will get two products. One, you will get three products rather. One, you will get earthworms, that is also a product. Then you will get the vermicompost, your second product, and then these jatropha plants will give you the jatropha seeds, which can be used to get the biodiesels. Then we are recommending one thing more to the farmers, that just give a 5-degree angle to the vermi bed which you make. So whatever liquid is there, that we are recommending the farmers to collect. Because of that angle, that waste can be collected here, and this is called as vermi wash. This is a very good quality, you can say, liquid manure that can be used by people for their crops. Then, friends, in northern part of India, we have number of biogas plants, lot of slurry is produced. So what we are suggesting to the farmers, that you take this slurry, do its vermicomposting in beds, and then means now when they do vermicomposting, one of their product is the biogas, second of their product is this vermicompost, third of their product is this earthworms. So like that, we are recommending them to have the maximum number of products from the waste, so that you can say benefit or profit is maximized. Then we are recommending this kind of channels to the people around their vermicomposting bins, because ants, they are a big enemy of these earthworms. So we want to stop the entry of these ants to the vermi bins. So we say you make one channel and put water into that, so these ants cannot cross this channel, so your earthworm will not be affected by that. Then we are recommending the one more second channel. This second channel is to collect all the liquid that is coming out from this vermi bin, that is shown here in the second photograph. We say collect it, and as I told you just now, that this is a very good quality manure, that is call it vermi wash, and it can also either be used by the farmers or it can be sold by the farmers. So friends, that's all for the today's presentation. I'm stopping this. In case you have any question, you can ask to me, friends. So over to the organizers. Yes.
Thank you, sir. Thank you very much. You have given very brief and very detailed information about this vermiculture, vermi beans, different species of earthworm, the physiochemical characterization of different compost. Really, it was very interesting, and our students, they are also interested doing such type of work for their research work, and that also undergraduate students. So I'm really thankful to you for giving such interesting talks. Muskan madam, do we have any questions from our audience?
No, sir, we don't have any questions as of now.
Yeah. Sir, definitely, but I'll be having many questions after listening your lectures. Definitely, I will contact you, sir, because our
Sure,
students,
sure.
they are interested doing such type of work. Thank you very much, sir, for giving
Thank
your
you, sir.
time.
Thank
And
you.
yes, sir. Thank you very much, sir. Yeah.
Thank you so much, sir, for sharing your knowledge with us. The next speaker of our, I would like to invite our next speaker, that is Dr. Sudipti Arora, scientist and assistant director at Dr. B. Lal Institute of Biotechnology, Jaipur. And I request Dr. Pagare to please introduce our expert.
Good morning, madam. I welcome you our international conference on current trends in waste treatment, reuse, and valorization. I'm very happy to introduce you. Dr. Sudipti Arora is an environmental research scientist and assistant director at Dr. B. Lal Institute of Biotechnology, Jaipur. Her specialization is in waste management, solid and liquid waste treatment, wastewater treatment by filtration technology, and other natural-based sanitation solution. She is the founder of Prakrit, a
Practice. Yeah.
center of excellence in environmental biotechnology, and she is also working as a director of the company. She had obtained her PhD from the Indian Institute of Technology, IIT Roorkee. She has also working as a guest faculty in Malviya National Institute of Technology, Jaipur. She is a member of International Water Association and has been involved in WASH projects from 2010. She has been involved in SARS-CoV-2 wastewater-based epidemiology research from India with other Indian partners and UK international partners through Global Challenge Research Fund. It is really great thing, madam, you are with us. She is also involved in the Indo-European projects titled Identifying Best Available Technologies for Decentralized Wastewater Treatment and Resource Recovery for India. It is what was known as the Saraswati, which was funded by DST, Government of India. She is also involved in an integrated technology intervention project funded by our DST. It is on transforming Andhi village in Rajasthan towards a zero waste model through green technology intervention. She is also guest editor in many journals like Water Special Issues, Pathogen Detection and Identification of Wastewater. She has authored and co-authored of important discoveries in environmental biotechnology in more than 20 science citation index expanded journals. She has guided more than 50 UG and PG students for their research. She has been honored by various awards, including Research Excellence Award 2020 by INSC for outstanding research work on filtration technology. And Dainik Bhaskar, they have given the Green Parrot Award in 2018 for her contribution in indigenous water treatment. Really, we are grateful, madam. You are with vast experience. You are with us. I'm very thankful for accepting our invitation for your today's lecture. And now it is over to you, madam. We are eager to listen. Thank you
Thank
very
you
much.
so much. Thank you so much, sir, for such a warm welcome and such a good introduction. I'm really grateful for this opportunity, definitely. So, my title of the today's talk is Potential of Earthworms in Removal of Antimicrobial Resistance and Antibiotic Resistant Genes, mainly during vermifiltration technology for domestic wastewater treatment. And nonetheless, Dr. Vinod, just before me, spoke a lot about earthworms and vermicomposting technology. So definitely it was a very great lecture by the sir. And I've also read a lot of research papers, and that gave me the inspiration in at first to start this technology. So at the very onset, I would like to thank you, sir. Uh, okay, just a second. Yeah. So, a brief introduction about me. Yes, I have done my PhD from IIT Roorkee and MTech from MNIT Jaipur, and that's where I started my research with. And with the onset of 10 years of experience now in vermifiltration technology, I've started my own NGO, which is called Prakrit Sustainable Integrated Foundation, that works for converting schools, colleges, and communities to a zero waste model, because I firmly believe that every waste is a resource, and as Dr. Vinod also said, that a lot of waste can be converted into resource. So the target is to convert each single waste into resource. Besides that, during COVID-19 we also worked on wastewater based epidemiology, where we carried out several researches on SARS-CoV-2 and to detect them from the wastewater as an early epidemic prediction. About my institute, Dr. Bilal Institute is a group of organization that is affiliated to University of Rajasthan, which was established in 2008. The focus area beside academics, research development, entrepreneurship, skill development, and holistic development. About the Prakrit, which is a center of excellence and an NGO. It is an umbrella center which is conceived by Dr. Bilal Institute of Biotechnology, Jaipur, that works on for fulfilling sustainable development goals. So, in my today's talk, I'll be speaking more on vermifiltration technology for wastewater treatment, the designs, features, our research insight from our research group over last 10 years. I'll talk about the mechanistic insights, that how earthworms actually help us in treatment, with a lot of significance and challenges and future research prospects. So, to begin with, yes, I know it's very unfortunate to see these pictures, but yes, that's the sad reality of India even today, that most of the sewage is still not connected to a sewage treatment plant in India, especially in rural areas, I'll talk about. So, there is an urgent need to develop an affordable, easy, natural, sustainable, and decentralized solution, because now we all know over several talks that we have heard on the topic that yes, there is a need still persisting to develop a technology that is sustainable in the long run. We don't require any technology that is treating wastewater but adding huge carbon footprint. So definitely we have to keep sustainable development goals in mind. With the same mindset, 10 years back, we thought of earthworm as an unheralded soldiers of mankind. They are the farmer's friend in true sense. They are the miracle organism. So considering the problem, considering this, we thought of as an opportunity to develop a novel, innovative green technology. Earthworms have been using, we were using earthworm over several years now for the treatment of solid waste. So we thought of using it for the organic portion of the wastewater. So there was a need to develop an efficient, effective, affordable, and robust technology that not only require less operation and maintenance, should also be very easy to manage, should also be very user friendly. So with this, we started working on vermifiltration technology 10 years back. We as the term says, vermi means we are using earthworms, and there is a filter which has different layers, so we call it as a vermifilter technology. Vermifiltration technology adapts the traditional vermicomposting into a passive wastewater treatment process using the potential of earthworms. This is the technology that involves the symbiotic and synergistic reaction association between earthworms and the microorganisms, which are not only present in the wastewater but also present in the filter media layers over the years, and also in the vermicompost. So together they react, they associate and process the wastewater, converting into nutrient rich water, that the effluent that we get, as well as vermicompost. So basically what happens is the body of the earthworm act as a biofilter. It allows mixing and aeration for the biodegradation of the organic matter through the enzymatic activity, and thus helps in the removal of contaminants. So this is a simple picture that shows that how vermifiltration technology work. So there's a filter. At the filter, we feed the wastewater at a very fixed hydraulic loading rate. It should not be too fast or should not be too slow. So we have already worked on that design part. I'll be speaking about it. And then we also use the organic fraction of municipal solid waste, as we know that it's their favorite food. So for the maintenance of earthworm in the reactor, we use cow dung as well as the organic fraction of the waste as their filter bed, as their dwelling habitat. And together, the specialized microorganisms which are present in the gut of earthworm, which are present in the waste, which are present in the wastewater, they together form an association. They form an activity together. They release certain enzymes, they break down the waste, and thus converting the wastewater into two main byproducts that we get. First is the treated effluent, which we can use mainly for agricultural application and other uses, garden uses, because it is highly nutrient rich, and also vermicompost as a manure. So over several years, we have been working on the different type of vermi filter. We started on from 2012 in IIT Roorkee when I was doing my PhD under the supervision of late Professor Renu Bhargav ma'am and Dr. Kazmi, where we started on with laboratory scale vermi filter. We used the basic plastic because we have no idea about whether they are going to work or not. So we just started with a very basic design, and we were not even able to access the real wastewater that time. So we started with the synthetic wastewater. Then over 1 year of study where we carried out different experiments, we got a good positive result from that, and then we moved to the pilot scale at IIT Roorkee, where we used both batch state and continuous vermi filters over synthetic wastewater. Then, after completing my PhD, I was shifted in 2016 to Dr. Bilal Institute of Biotechnology, and here I joined as a research scientist. And then we started on with the field scale. So we really needed to check whether the whatever happens in the lab is actually working in the field scale or not. So we started with the actual wastewater this time, the institutional wastewater, and then lot of researches have been carried out later. So these were the initial pictures of the vermi filter that we set in 2012, where we call it as a jugaad system, because that time funding was also not available much. So we were not even sure of that what will happen. So we started with a basic plastic containers and just the simple experiment to see whether what we are thinking is going to be working or not. So also we tried to attempt different runs this time, where we worked on different parameter of the design of vermi filter. So first very important parameter was stocking density. We wanted to check whether what how many earthworms are required to actually treat the wastewater, because it's very important that the number of earthworm, the weight of earthworm, the health and maturity of earthworms. So these were different parameters which are really important if we design vermifilter. So we carried out experiments with different stocking density from 8,000 to 25,000 worms per meter cube, and we were able to find out that in terms of BOD COD removal and pathogen removal, that optimum stocking density is 10,000 worms. So it's very important that the number of earthworms should not be too high, because then the food will be less. So we have to maintain the food to mass balance ratio. Then second question was which filter media to use, because in earlier studies we found that gravels were the most cost effective material, and they're easily available, as well as they are good. But then what is the size of gravel, and they should be smooth, they should not be very rough, because that will kill the sensitive earthworm species. Their skin is very delicate. And we also thought that let's also use coal, glass ball and mud balls. These experiments were also conducted. We thought glass balls would be very smooth, so maybe earthworms would love that habitat and will help in increasing the treatment efficacy. And coal we thought of because that would also help us in removing the color as an activated carbon replacement. So we did different experiments, and definitely we got good results in all the cases. But definitely the top material that we came on conclusion was the riverbed again, because definitely it is cost effective also and has much better advantages. Then the question was to see different hydraulic loading rates. How much water is required? We cannot have a lot of wastewater at a very high flow rate, because that would disturb the vermi filter and would also do the clogging. So it is very important to maintain HLR, HRT, OLR. These all ratios were maintained at this stage, and we found from zero point 0.5 to 2.5 meter cube per meter square per day, the optimum HLR came out to be 1. So based upon this, the final design criteria was established. And then yes, the treatment efficacy was also important. We have to check BOD COD, nutrients and pathogen removal. So we came up then with the second phase of our experiments, where we started with the pilot scale vermifilter. Then this time we replaced our plastic with perspex material. That's another kind of plastic, but this is more sturdy one. And then we divided this into two parts. The first part was the one with only the filter media, that's a control, and we did not add any earthworms. And the second part was the vermi filter, where we actually added earthworms, just to see that how much advantage this earthworms are giving as compared to a normal filter. So we named it as biofilter and geofilters. And we did this experiment for 120 days, where we used OFMSW from the canteen waste, synthetic wastewater this time again, and Senna fetida species. Yes, we also did experiments with different earthworm species, Eudrilus, Perionyx excavators, and Asena fetida. But again, Asena fetida is the most sturdy one and was easily available, and it gave us good results as well. So we stick to that, and then we performed this experiment where we saw that pH was definitely neutralized after adding earthworms to the vermifilter. Temperature was roughly maintained at the room temperature. DO was considerably increased. That's a very good part, that this system was completely aerobic. This gave us good hope that there is no smell coming out of our vermifilter, which is again another indicator that our plant is working fine. System is aerobic, and earthworm has this beautiful capacity to dwell within the habitat and do the burrowing activity. So that gave us the aeration of maintained the aeration of the vermifilter. And definitely BOD percentage was 93% removal was observed, and 73% COD removal was observed. So this was the experiments we did. And we also, yes, so for the first time our group reported the removal of pathogens during vermifiltration. So far there was no report on removal of pathogens. People mainly focused that time only on BOD COD, but we focused on the pathogens because it was very important to again look at the pathogens. If we have to reuse this water for any purpose, even irrigation, we have to maintain the FC fecal coliform and other parameters. So around 3 to 4 log removal was observed, and our FC was within the standard range. That time the standard range was 1000 mpn per 100 ml. Then we started on with the third phase, where we used OFMSW again, and the real wastewater, to see what happens when we use these. So again, and we also saw very interesting result that the OFMSW after 90 days, we took the sample after every 30 days, and we saw that this has been converted into good vermicompost, as you can see by the CN ratio and other parameters. Pathogens were also considerably removed, and the nutrient ratio was maintained. So we got good vermicompost as an harvest, and microbiological parameters were also maintained. Now, this was very important that we need to further investigate the mechanism. No technology will grow if we do not do the R&D behind that technology. That is very important. So one thing was sure, that earthworms definitely helped us in removing the contaminant, but we also cannot ignore the importance of the microbes that are present within the vermifilter, that is enhancing the process, because it is ultimately microbes that are working behind the treatment. So we investigated what all microorganisms are present in different layers of vermifilter in the earthworms, and we also did the molecular analysis and made the phylogenetic tree. And we also saw that mainly the group of bacteria was belonging to two phylogenetic group, gamma proteobacteria and firmicutes. So now then, this time we thought of going to this another phase of our study, where we have installed this vermifilter in a field now, with actual institutional wastewater from Dr. Bilal Institute of Biotechnology. Also, we also did this for the first time for clinical laboratory. So Dr. Bilal Institute of Biotechnology has an industrial partner here in the campus, where all the diagnostic and pathological tests have been done. So blood tests, urine tests have been done here, and it was presumed that the sewage that comes out is of clinical nature, is of highly pathogenic, and no such reports have been done for vermifiltration. So we wanted to check it for the first time. So we installed twin vermifilter now in the campus. There were two vermifilters. One was for the institutional wastewater, and second one was for clinical laboratory. And this plant was run for one entire year, and a group of lot of students have done their thesis here for this project, MSc thesis. And then we were able to see that the the final effluent was really good in quality, within the parameters of the standards. And we were able to decipher that yes, vermifilter does well in the field as well. We just need to keep certain conditions, like we have to give them shade. They cannot be in direct sun, they cannot be in direct rain. So these are the pictures of that vermi filter we installed in the campus. Luckily, we also have a vermicomposting unit here. I'll show you the pictures. So we maintained the vermicomposting unit also since then, and Acena fetida is available here. So this we are like harvesting these waste. We are also doing composting, vermicomposting here. So we had that thing in mind. And this is the final design of this vermi filter, where we are using gravels of different sizes, sand, and vermigratings, and organic fraction of municipal solid waste. So again, this time we thought of investigating the microbial diversity along with their properties. So we did the bacterial diversity of different uh, units, like from the influent. We wanted to see that which is the dominant strain at which level. So influent, effluent, earthworms at different phases of their life. So there were some earthworms that were not fed in the vermi filter, so that we call them as control earthworm. We wanted to see that any changes happen within the earthworms or not. So we harvested earthworm after 180 days, then 360 days, and we also extracted coelomic fluid from the earthworm. So this all experiments were done to see that what is the actual mechanism behind the vermi filtration technology. Coming to what is the key differentiator, why vermifiltration technology? I always abide by this technology because this technology is definitely cost effective, user friendly. If we go for rural, we talk about rural India, so it's very easily understandable by the people, because they have been working for with earthworms for over several years. So this does not have any hi-fi operation, so this is very user friendly, green technology. With Because the entire system we made here is without electricity. So we are just pumping one time sewage from one institute together at a septic tank just once, and then we are making it flow under gravity. We are maintaining HLR through gravity. So we say it's a green technology, unlike other commercially available technology where huge carbon footprint and electricity is required. This technology is definitely maintenance free or repair free, because very little maintenance is there. We just need to harvest the vermicompost from time to time, and that even a unskilled labor can do. So that is very easy technology. The consumables that are required in this technology, like all the filter media, is very cost effective, does not involve huge cost, very easily transportable, voted, and you and we can use the locally available things, and also it gives us the useful byproducts. Why And then now coming to what is the novelty factor of our vermi filters, how it is different from other vermi filters which are already available in the market. So we have designed this vermi filter in such a way that they are bioengineered and customizable according to the user centric requirement. So as per the design, we customize it. It has a unique and specialized design which is unique to us. We have already applied for the patent and has covered two clear 2 rounds for that, and it's in the last phase. We have also our own specialized microflora now, since we are working on the microbes. So we design, cultivate our own specialized microbe, and then put it into the vermi filter. And definitely we have worked on different segment and still doing a lot of research on different segment, like textile industry, hotel industry, pharmaceutical industry, and hospitals. So it's at a R&D stage, but very soon we are going to publish some data on it, that it can be used at multiple sectors. And definitely it's scalable from 1 KLD, a very small unit, to 1 MLD. These are the pictures of the vermicomposting unit that is here at our campus, and we are maintaining it, and we are also adding garden waste, kitchen waste into it. Now coming to very important point, because while we were working on these technology, so very important factor was that why this technology has an edge if we talk about the hospital wastewater or clinical laboratory wastewater. So the one thing that was very fascinating to us was the global problem of antibiotic resistance, which has become even more prevalent now after COVID-19. Because as we know that during COVID-19, a lot of indiscriminately antibiotics have been served, whether it's a viral, but we were having antibiotics at a very and that mindlessly, I'll say, doctors even prescribe. So what will happen and what is still happening is the antibiotic resistance. So here, during vermifiltration technology, we really wanted to see that what happens to the antibiotic resistant bacteria. So we chose different classes of bacteria, antibiotic belonging to penicillin, cephalosporin, aminoglycosides, and others, which have their fixed mechanism of action. So what we saw was that we compared the bacteria, antibiotic resistant bacteria, in influent versus effluent, and to our surprise, we saw that considerably the antibiotic resistant bacteria were reduced in the effluent. So that means there is some mechanism that is going on which helps in removal of the antibiotic resistant bacteria during vermifiltration technology. To further prove it, and to go into details and depth of this, we performed the molecular activity of these antibiotics. So what we did was, we took the genes which are responsible for particular class of antibiotic and did the PCR analysis. So for example, for ESBL, there are three genes responsible: SHP, TEM, and CTX. So we did the PCR profiling and we saw that SHP and TEM were absent and only CTX was present. So that means this particular gene is responsible for resistance in our case. Similarly, we did for MRSA and colistin, these three classes of antibiotic. So these were the results of the page agarose gel electrophoresis, where we saw the presence of gene. These all studies, these results, what gave us these results actually helped us in elucidating the mechanism, that how is this possible, that this technology not only helps in the treatment but also reduces the antibiotic resistant bacteria and antibiotic resistant gene. So we came to the conclusion that in the vermifilter there is this ecosystem of earthworm and the indigenous microbes. Now we know what microbes are already present because we did the molecular analysis. So what actually happen is that these earthworm and microbes make an established film. We call it as a biofilm. And in this biofilm, breakdown happens: physical breakdown, chemical breakdown, and biological breakdown of different contaminants. And when these contaminants are exposed to this film, so what happen is the antibiotic resistant bacteria gets reduced. And so that's why they are very less in effluent. So this whole process is a little, definitely we are still figuring out at a later stage with more advanced technologies, but to such extent we did this deciphering the molecular mechanism. And also what happens inside the earthworm for this. So we have already published the papers on this, that there is an interaction between earthworm and microbes to actually treat the wastewater and reduce ARM and ARGs. Further, one very interesting result also we did was to see what is the effect of this finally treated water on the plants. What actually happens, because ultimately no technology will be useful if we do not actually use our treated water for irrigation or maybe some other purpose. So because we have a beautiful garden here at the campus, we thought of now using this water actually in the field and see that do they really affect the seed germination and the seedling performances. So we did different experiments and plant we also have a plant biotechnology department. So this department voluntarily took this from us, from the environmental department, and they did all this study, and they compared that the germination and seedling ratios and all those things with these things like the normal tap water, the untreated wastewater, then the treated wastewater with dilution and without dilution. So they saw that with 50% dilution, 86% highest percentage was there in germination as compared to treated water without dilution. It's almost same, but then tap water gave less. So this was a very interesting finding for us, that vermifilter effluent definitely gives us even better quality of, better quality irrigation quality water, so that our plants can be harvested and germinated well. So we did these experiments on the field scale also to see this germination and other properties. Also we are exploring more now on the enzymatic properties and other things. So these all lawn is maintained by the vermifilter treated water: root length, shoot length, seedling length, and other parameters. So we have been working on this, and this paper is now in writing. And this was the last set of experiments to further see the molecular mechanism of earthworms. For this, we took earthworm from different sources, like one from the Goshala, one from our own vermicomposting unit, one we harvested from our vermi filter at different phases. And then we extracted coelomic fluid from this by cold shock method to see the protein analysis. What is the protein content present in the earthworm? And so these were from different, like from Goshala, from clinical vermifilter, from domestic vermifilter. And we did this experiment to see the bands. Basically, this is the SDS page to see the protein bands in different set of earthworms. And very interesting, we saw that the uh, original earthworms, like those from the Goshala, which were not from our vermifilter, have certain bands which protein bands. And the earthworms, after 45 days and 80 days after we added it, showed the same band. That means that once the vermifilter once the earthworms get into vermifilter, they get some time to adapt. They immediately may not respond. Sometimes they even get shock, because even they are natural live organisms, so they get shock. But after some times, they do get acclimatized, and that is shown by these experiments, that they take several days to acclimatize, but once they are acclimatized, they showed the same property as the original one. So these were the preliminary findings, and still we are figuring about establishing some more environmental interactions with earthworms for a long period of time. And these are our future and ongoing projects to understand vermifiltration technology in detail. And not only we are doing the continuous R&D, we are also in talking terms with several institutes, industries, and rural villages to have this technology in real scale. So we have recently we visited this village Andhi in Rajasthan, where we saw that there's a lot of problems concerning to the wastewater as well as solid waste. As you can see, lot of solid waste is accumulated, and that clogs the ponds. So definitely there's a huge problem. So with this, we applied for a project to DST for transforming this Andhi village to a zero waste model. And happily, I would share that we got that grant. It's a 3 crore project where one of the objective would be to install vermifiltration technology for this village. And definitely we have recently received the fund also, and next month from March we are starting on with this project. So maybe very soon I'll be sharing more about it. And these are the different partners involved in this project. And we are now having this technology on board from the research to finally at the implementation state in one of the village. So this gives me immense happiness, because we are not only fulfilling the sustainable agenda 3 and 6, but doing much for the environment. Thank you so much.
Thank you, madam. Really, you have given very good information about the novelty and R&D behind the vermicompost and vermiculture. Uh, I like the important thing that you have mentioned. You have talked about the microflora, and which is very important topic, and we cannot neglect it at any point of time.
Yes.
Because microflora is very important when we are adding, because this adds the nutrient in the soil. And but I have one question, madam.
Yes.
When you treated the sample, this thing, vermicompost and your clinical sample and your normal sample were there, did you check the vermic filtrate with the characteristics? So it is showing the some differences in seed germination.
For clinical laboratory, no, sir, because ultimately,
Yes.
after going in from the vermifilter, they are giving us the good results in terms of the nutrients. But yes, one thing we noticed that at one point of time there were some toxic material present in the clinical laboratory. So that
Huh.
time, few of the plants did get like they were not germinated. So we then find out
Okay.
that whenever there is any toxic material coming from the clinical laboratory, maybe because of urine test and blood test, that time it may not. So we added, then at that time we thought of doing the post treatment. So we did this experiment of adding chlorine to the
Okay.
wastewater. So like
Yeah.
we did just very small ppm. So we did from 1 to 5, and 1.5 ppm of chlorine gave us the good results, and that then we can do this for you. You can use it for irrigation.
Okay, okay, okay. Thank you, madam. Also, you have involved all your molecular techniques in your study also, though it is conventional work, because we say vermicompost, anyone can do it, but you have involved all the phylogenetic study also. You have studied the microflora with the phylogeny. Really, it was wonderful work, and since you got the 3 crore rupees of grant of on this same topic, really, we congratulate you and thank
Thank
you,
you.
madam, for sharing your all the information and all. Thank you very much.
Thank you so much. Thank you so much.
Excuse me, ma'am. There's one question from the audience. Hello.
Yes, ma'am.
Ma'am, actually, the question is, how can we reduce the time required for the adaptation of the earthworm?
We are working on it. So for now we need at least a month for acclimatization. That's what we saw, because initially bod cod and everything gets increased when we are adding earth form, because earth forms are in a stress. So even they start releasing certain material that is adding on to bod. So 30 days minimum is what we think, but we are still trying to research upon that what can be done. But I think 30 days is like not that much also. Once we establish the technology, and
Yeah.
it has to go a long way. So it will go for 5 years. So 1 month is simply very less time for them to. After 1 month they start working, they get acclimatized, and then after that they work beautifully.
Okay. Thank you so much, ma'am, for such an insightful
Thank
presentation.
you.
Thank you.
Thank you.
Next, I would like to invite our next speaker, that is Dr. Ritu Singh, Assistant Professor in the Department of Environmental Sciences, School of Earth Sciences at Central University of Rajasthan, Ajmer. Now, I would like to request Dr. Pagare to please introduce our expert. Over to you, sir.
Thank you, madam. I welcome Dr. Ritu Singh in our international conference. Uh, Dr. Ritu Singh is working as assistant professor in Department of Environmental Science, School of Earth Science at Central University of Rajasthan, Ajmer. She has received her PhD in environmental science from Indian Institute of Toxicology Research, CSIR, IITR, Lucknow. Her doctoral studies involved synthesis, characterization, and application of zero-valent ion nanoparticles for remediation of organic and inorganic contaminated soil and water. At present, her research group is engaged in developing mitigation methods for endocrine disruption chemicals and emerging contaminants. She has been considered the experience on in the area of pollution monitoring, remediation of environmental contaminants using nanoparticles. She has published two international books. One is with Springer and another one is the Taylor and Francis. She has been awarded with Young Scientist Award in the field of environmental science by the Lela Education Foundation and Academy of Environmental Biology. She is also editor of the journals like Journal of Nanoscience, Nanoengineering and Application, Journal of Water Pollution. She is also working as a reviewer of many journals like Chemosphere, Journal of Hazardous Material and all. Also, she is also the reviewer of the PLOS One, Applied Water Science and all. Uh, I really welcome, madam, for your expertise in your field. Now, over to you, madam. We are eager to listen you. Thank you very much.
Thank you so much, Bakari sir and Muskan ma'am. Am I audible? Please let me know if
Yes,
my
ma'am.
voice is clear.
Yes, ma'am, you're audible.
Thank you, ma'am. Thank you, and thank you to all the members of organizing committee of this international conference for giving me this opportunity for sharing my research in this platform. So quickly, I'm going to share my screen. Just let me know if it is visible to you. Is it visible to you, ma'am?
No, ma'am, it's not visible yet.
I hope now it is visible.
Yes, ma'am, it's sharing.
You can see now.
Yes, ma'am, it is visible now. Yes, ma'am.
Okay, okay, okay. So today I'm going to um, talk about the application of nanotechnology in environmental remediation. And this nanotechnology is particularly is the creation of useful or the functional material devices and the system to control manipulation of matter on the nanometer length scale and exploitation of the novel phenomena and properties which arise because of the nanometer length scale. Now, these nanometer materials they exhibit unique physical, chemical, electrical, and mechanical properties, which actually offer potential beneficial application in industrial sector, pharmaceutical industries, then food processing units, communication system, textile, everywhere. So, in recent years, the use of engineered nanoparticles, nanomaterial, they have gained increased amount of attention as potential remediation techniques. Now, these engineered nanomaterials, these are generally defined as the materials with size range of approximately 1 to 100 nanometer, and these nano-sized material they display different properties compared to their bulk counterparts. And because of these novel properties at the nanoscale, a range of these engineered nanomaterials these are used in a number of consumer products and other applications, including the environmental remediation. So, nanoremediation is basically the term which is used to describe various techniques and the methods to clean up the contaminated site using engineered nanomaterials. Now, according to one data, it has been estimated that there are between 50 to 75 sites around the world that have used these nanoremediation techniques either at pilot scale or full scale study. Now, next here, this is showing the multimodal characteristics of nano remediator towards environmental pollution. So, you can see here that filtration or this nano remediation could be carried out. There are too many processes: filtration, chemical reaction, oxidation, reduction, co-precipitation, then photocatalysis through nanocomposites or nanoparticles, absorption through nanotubes or nanoparticles, and there are sorbents, sponges, metal oxide nanoparticles. So, because of this multimodal character and excess of nanostructures which are available, each category of contaminant may potentially find its remedy. Say, for example, inorganic cation and anion, because of their charge, they are prone to complexation and absorption on the surface of metal oxide nanoparticles, or maybe carbon or polymer based nanoparticles. Okay, and say, for example, remediation of redox active inorganic anions or maybe radionuclides, they require reducing conditions. So, in that case, zero valent metal nanoparticles of maybe nickel or copper, iron, they could be applied. Similarly, then chlorinated compounds like dioxins and herbicide, insecticide, PCBs, these are well suited for chemical reduction. And polycyclic aromatic hydrocarbons, they can be well degraded by oxidation reaction. So I will come back to nanoremediation again. Here I am highlighting the contaminant which I have selected for checking out the efficiency of these nanoparticles. That is dyes. Okay, and one of the reason of particularly selecting the dyes because here in Rajasthan, textile industry is this is one of the most important industries with regard to production and the employment. And here in Rajasthan, Pali has the highest number of textile industry. It is around somewhere 1,000 industries are there. Similarly, Sanganer has almost 600 textile dyeing units. Then Bagru has 250 textile dyeing units. So the wastewater which is generated from these industries that is huge and it contains mixture of undesirable dyes along with varied contaminants posing serious threat to the global environment. And it is stated that about 15% of the dyes is reported to be released directly into waste effluent every year by textile industries. And we are very well aware of this thing that these dyes are highly resistant to natural degradation methods and their presence in the aquatic system that could have an adverse impact on the human health as well as ecosystem. They are also known to exert their toxicity even at very low concentration and they are carcinogenic in nature, they have mutagenic, teratogenic effect, they are also listed as emerging contaminants. So in the present study, actually, two synthetic dyes have been used as a model contaminant. So one is crystal violet, which is a synthetic catalytic dye that has broad array of industrial applications like food staining, biological agents, food additives, medicines, textile, leather industries. Another one, this is this is anionic dye, which is reported to be used extensively in mining activities and pharmaceutical industries and textile manufacturing units. So both of these dyes, these are toxic and they pose significant threat to the aquatic life, human being, and they disturb the ecosystem services as a whole. So therefore, to eliminate the hazardous impact of the dyes, it is very much required to develop a competent technique that can remove dyes successfully from the water matrix. So several treatment methods have been employed over the years to treat these dye-contaminated wastewater coagulation, propagation, oxidation reduction. Among them, that adsorption that has become a hotspot in wastewater treatment because of several advantages: simple design, then cost effectiveness, and flexibility of application. So several, in this regard, several nano sorbents have also been applied for the removal and the separation of pollutants, particularly I'm talking about dicontaminants. And among these iron-based magnetic nanoparticles, these are one of the highly researched domains for their great potential in pollutant remediation. And among the most widely used nanoparticles is zero-valent iron nanoparticles and magnetite nanoparticles, magnemite nanoparticles. They have been increasing interest in the area of environmental remediation. FeCO4 nanoparticle has shown significant potential for the remediation of several pollutants since it shows both absorption and reduction at the same time, which make it a better candidate than FeCO3 nanoparticle, which shows only adsorption process. The same thing has been shown here. Anxiety also is a better remediation technique than FeCO4 nanoparticle because it undergoes reduction, flow precipitation, and absorption at the same time. But it is highly unstable in nature, and as soon as it exposes to the air, it oxidizes and it agglomerates, which decreases its potential as a remediation agent. So here is some advantages of using the iron oxide nanoparticles that have been presented, which include high absorption capacity, enhanced reusability, high selectivity, low cost, better stability, less time consumption, and then ease of separation because of their magnetic behavior. So all of these traits that increase their suitability for selection. But there are some constraints. The ability of iron nanoparticles to get oxidized and lower selectivity for some of the contaminants in water matrices that has limited its removal potential to some extent. And beside this, there is a need of air restricted setup or maybe continuous purging of nitrogen during the synthesis and remediation process as well. So we prepared one air stable iron oxide nanoparticle, glutathione functionalized magnetite nanoparticle that can be actually synthesized with controlled use of an modifier, eco-friendly modifier, without the need of any complex setup or nitrogen purging. So the synthesis of this magnetite nanoparticle was attempted through a chemical synthesis route, that is oxidative etching-based wet reduction method through controlled use of glutathione. Glutathione has been utilized here as a modifier. So this oxidative etching is actually a natural phenomenon which is responsible for a general rusting of steel material or iron material whenever they are exposed or subjected to air and water for long term. Okay. So that process has been utilized here for the synthesis of this functionalized magnetite nanoparticle. So the process is very simple. Here you can see in the presence of oxygen, FeCO4 nanoparticle they can be engineered well by regulating the oxidation process through careful use of glutathione. So the process generally involves addition of ferric chloride to glutathione under certain set of experimental condition and then reducing it with sodium borohydrate. And the appearance of black color that indicate the presence of nanoparticle when it is washed and then separated magnetically for further use. Then their nanoparticle has also been prepared just for the comparison purpose in the same manner, except the addition of this glutathione. Now, This glutathione, which has been selected here as a modifier, this is a natural polymer that comprises of different functional groups like thiol group it is having, then carboxy group is there, amine group is there, and these functional groups they attach to the surface of nanoparticle, and that attachment prevent them from the agglomeration, and it increases ultimately its stability, making this nanostructure more efficient. And at the same time, glutathione it controls the nanoparticle size and keep the magnetite nanoparticle mostly monodisperse in nature. So once the entire synthesis was done, these were characterized using multiple techniques. First was DLS, dynamic light scattering. So the particle size of modified magnetite nanoparticles that was analyzed by this DLS, and these two figures shows the distribution of bare and modified nanoparticle. So here in this figure, this is showing the size distribution of Fe3O4 nanosorbent without modification, and this is indicating like large and heterogeneous size of the absorbent. One peak is here at around 1 to 4 nanometer, one peak is here, and the approximate size is around 193 nanometer. Where on the other hand, the modified nanoparticle, if you see here, you can see more uniform, the smaller size of absorbent. So the hydrodynamic diameter of this modified magnetite nanoparticle that was around 24 nanometer, and the polydispersity index that was also came less than one, which indicates that the particles are more uniform and more dispersed in nature. So that is the purpose of using a one modifier for the synthesis. Then this is showing the X-ray diffraction peaks of the modified nanoparticle. So this characteristic peak at 235 degree, this is indicating the presence of iron oxide phase Fe3O4, and this Fe3O4 also corresponding to the extrinsic planes, with this 220, 3114, and so on. So these diffraction peaks that actually were in accordance with the JCQBS card. Now, the difference between this XRD knots of bare and modified nanoparticle is that this bare nanoparticle it shows noisy peaks, whereas this modified nanoparticle they show sharp peaks that confirms the good crystalline structure of the modified nanoparticle. And since no separate peaks were formed, it can be concluded here that the formed nanoparticle these are single phase crystals without the presence of other phases of iron oxide, maybe FeO or Fe2O2. So that was completely absent. Then later on, some images were obtained for the magnetite nanoparticle. This is for bare and this is for modified magnetite nanoparticle, and these are the after absorption images of these modified magnetite nanoparticle. So here in this case, you can see this bare Fe2O4. This shows a spherical shaped morphology with very high degree of agglomeration. It is forming clusters and doesn't have any definite pattern. Whereas here we have modified NP showing formation of some chain-like nanostructure. So here we can see that the presence of this glutathione, it acts as a cross-linker and itself assembles the magnetite molecule into long chains of nanoparticles. So these are some of the after absorption images of Syrian PR dyes. And here we can see the deposition of dye molecules on the surface of nanoparticles. And this is eliminating the sharp structures and defined shapes of nanochains you can see here, right? And also these two figures this shows the irregular and bulk attachment of CV dyes and PR dyes. So this was further confirmed with this EDX analysis. So this figure shows the elemental composition of this glutathione modified nanoparticle and confirming the presence of iron and oxygen of Fe3O4 nanoparticle, the presence of carbon and sulfur that indicates the coating of this glutathione on the surface of magnetite nanoparticle. Now these two images, these are the after absorption images, and in both the cases there was an increase in carbon and oxygen percentage, while iron percentage that was observed to decrease. Now besides the presence of these smaller peaks of this nitrogen and chlorine that confirm the attachment of CB to the surface of nanoparticle, while the presence of this sulfur here that confirms the PR dye absorption. And the last characterization that was done with FTIR, and here in the presence of 502 and 64 band that corresponds to the stretching vibration of FeO and the presence of magnetite in oxidized form, the absorption band at 3431 that indicate the stretching vibration of hydroxyl group present over the surface of nanoparticles. And this is representing the after absorption images. This is for CD, this is PR, and this is methylene modified nanoparticle. So here in the absorption peak at 1630, that indicates the energy stretching vibration of any group of GSH, coated affecting open nanoparticles. Whereas in case of this CP, the CC stretching vibration that hits the absorption peak at 1500, indicating the presence of aromatic ring structure, and the absorption peak at 1377 and 1172 that represents the CN stretching of tertiary engine present in CV lines. Similarly, in this case on PR, the absorption peaks at 1385 and 1351 that represents SO stretching vibration of circulate group that is present in PR dye. There are other peaks also that much detail I have not given here. So this ultimately indicate that both the dye CV and PR get absorbed over the surface of magnetite nanoparticles. Now, after characterization, we studied the effect of different reaction parameters, and one among them is absorbent dose, then pH, we have studied temperature, and initial concentration we have studied. So, as here in this graph, you can see the dose of the nanoparticles that has been increased from 0.7 to 2.5, and the absorption efficiency, it increases along with the increase in dose. And this is showing an excellent removal capacity somewhere from 25% to almost 100% in both the cases, and that is a 30-minute time interval. Now, the higher amount of adsorbent dose increases the number of surface active sites which are present in the aqueous solution, which absorbs the dye molecule more rapidly. But one thing has to be noticed here: the absorption capacity is scales up with a lower amount of adsorbent dose, as a large number of dye molecules get attached to the entire surface of nanoparticles more steadily. So, when their quantity is low, so a local dose of only 1.8 that has been utilized for further experiments. As far as this pH is concerned, so because the CV and PR dyes, one is catalytic and another is anionic, so they show opposite. So here in the lower pH that actually favors the removal of anionic PR dye. Since PR dye has high negative charge density and the dye molecules at lower pH, they tend to get attached to the surface of nanosorbent because of excessive presence of positive ions in and around the nanoparticle. But in case of CV, the removal efficiency gets escalated at higher pH range. So when the pH was ranged from 3 to 11, the increasing number of hydroxyl ions and other negative ions created an electrostatic force of attraction between this positively charged CV molecules and the negatively charged adsorbent. So this in turn helped in rapid CD dye absorption. So the maximum removal of CD and PR dye that was observed at pH 11 for CD and pH 3 for phenol red dye. Similarly, an increasing trend has been observed in case of temperature also, with an increase in temperature from 25 to 65 degrees Celsius, the absorption efficiency increases. Now, the reason may be ascribed here to the rigorous motion which is created between the dye particles and the adsorbent molecule at high temperature. Okay, so this results actually in the building up of activation energy, which accelerates the absorption reaction. And this process in turn, it raises the removal efficiency of the magnetite nanoparticles. And in the other ways, it can also be, it can be uh, stated that temperature increases actually the mobility of the dye molecules and making them well dispersed in the solution, which then actually amplifies the degree of interaction between the uh, absorbent and the absorbent, and finally boosting the dye uptake. The last figure here you can see this is the effect of initial dye concentration, and the removal rate of nanosorbent it tends to decrease gradually as the initial dye concentration increases from 100 to 500 mg per liter. So initially the dye absorption was fast, but then gradually the absorption decreases when the absorbent reaches their equilibrium point. So the basic mechanism involved here that is the availability of large number of active sites on these linkage that actually quickly bond with the dye molecules at the beginning, but with the increasing time, the reaction rate it becomes slow as the number of surface active sites reduces and the concentration of dye molecule increases in the aqueous medium. So finally, the absorption stops when magnetite nanoparticle it reaches their saturation point, or you can say when it reaches their equilibrium points. Then later on, this absorption kinetic was evaluated using zero first order, second order, and then diffusion model. So first order reaction model that gives a linear coefficient for CV and PR dyes, that is around 0.95 for CV and 0.91 for PR, and this reaction model fitted well with the absorption rate of more PR dye, which clearly shows that the physics option that has taken place as a chief mechanism in removing PR dyes. Whereas in case of CV, the second order model that gives a straight line for CV dye with a high regression coefficient 0.98, while PR dye shows a little bit lower values 0.77. So the absorption data for CV, well fitted to the second order reaction model, this indicates that chemisorption has taken place in ribolic CV. So for PR it is physics option, for CV it is chemisorption basically, the dominant mechanism. Then intraparticle diffusion that is also used to analyze the absorption mechanism of adsorbate and absorbent reaction. And the fitting curves for CD and PR dye that is displayed two phases here. Um, which can this can be used to describe different types of absorption process happening in the reaction system. So the first phase of fitting curve for the dyes that depicts the instant adsorption of the dye molecules on the nanoparticles. Whereas the second phase, this describes the intraparticle diffusion happening. So whenever the linear plots pass through the origin, the chemical reaction is considered to be controlled entirely by intraparticle diffusion mechanism. Right? But because in this graph, both the linear segments of CV and PR dyes do not pass through the origin, so this suggests that there are more mechanisms involved in the absorption process besides intraparticle diffusion. So this table is the values of different parameters of these kinetic models have been calculated, and the same thing has been given here. Then this absorption isoforms, this actually studies the manner in which the dye molecules get absorbed on the surface of these nanoparticles. So here in three isotherms, Lanier, Frenlich, and Temkin isotherms that have been used to fit the experimental data obtained from dye absorption studies. So Lanier isotherm model fits well for the absorption rate of phenol red dye with a high R2 values 0.99, and for CV also it is very high 0.95. So this indicates maximum monomer absorption, right? The R2 value for this regression coefficient value for crystal violet and phenol red dye in this friendly isoform as well as in this Temkin isoform, these are found to be quite low, and this both of these did not fit the straight line. So this monomer absorption that was the main process, main mechanism, and the experimental values and the calculated parameters have been presented here. Later on, the thermodynamic parameters that were also calculated, and here is positive value for both CV and PR dyes of the enthalpy. This indicates that absorption reaction is mainly endothermic in nature. That means the energy is actually consumed during the absorption process. And this could be because of the reason that the dye molecule has to lose a fragment of their hydration cell in order to get absorbed on the surface. And this delta T, the positive value of delta T indicates the feasibility and spontaneity. This negative value, sorry, negative value of this Gibbs energy that indicates the spontaneity of the reaction, and this positive value of entropy that implies increased degree of randomness at solid liquid interface. Then there can be three possible way of interaction between the dye molecules, whether it is a cationic or the anionic type, and the nanostructure causing the absorption reaction. Now this could be simple electrostatic force of interaction, between the anionic functional groups of nanoparticles and cationic dye molecules. In case of anionic pH dye, an acidic pH environment that is required to accelerate the absorption reaction. Another mechanism is hydrogen bonding. Hydrogen bonding, which may occur between the nitrogen atoms of CD-dye and hydrogen species of the functional groups of these GSH modified nanoparticles. Pi interaction could be there between the benzene rings of the dye molecule and the cationic nanoparticles. Basically, this kind of interaction, it takes place between pi electron and cloud interact with the cationic or anionic molecule, just as in the case of electrostatic interaction. Lastly, this the application of the entire nanoparticle which has been synthesized, modified nanoparticle that has been investigated on real samples, and these were subjected to three different types of water matrices. One is tap water, then sewage and textile industry, and the findings showed an excellent removal efficiency of these modified nanoparticles even in real water environment. So nanoparticle they showed highest efficiency that took water matrix with 92% and 70% removal, while 90% and 61% removal that was observed in sewage samples. In textile industry, comparatively lower removal rate was noticed, and that is because of the difference in the matrix, the composition of the matrix. Tap water generally has lower amount of dissolved impurities, whereas these STP water and the textile industry wastewater, they have lot of organic and inorganic contaminants which actually compete with the dye molecules for the surface of for the reactive sites of nanoparticles, and thus it decreases their uptake capacity and removal efficacy as a result. So overall, the conclusions are there. The oxidative etching, which has been shown here, that favors the growth of these magnetite nanoparticles to control addition of glutathione during synthesis. This glutathione used for surface functional isolation that increase the functional group mobility, molar viscosity, and stability. Both for CV and PR, these modified nanoparticles showed excellent absorption capacity of 1690 and 1360 mg per gram. And CV removal, it fits the first order kinetic model, while the PR dye removal fitted well with the second order kinetic model. The adsorption reaction fits the Langmuir isotope for both the dyes, indicating the monolayer adsorption. So, briefly, all of these findings, they indicate that this kind of functionalized magnetic nanoparticle, this is a promising nanosorbent which could be employed as a better alternative for the management of not only the dyes and the textile waste contaminated site, but also for other kinds of organic contaminants, because these are not contaminant specific nanoparticles. They can be well employed for other kind of contaminants, environmental pollutants as well. But there is one concern that in order to increase their employability in the water treatment system, the possible degradation product of the dyes and their associated risk, they should be investigated further. There are some concerns over the toxicity of these nanoparticles. There are some concerns over their tactical applicability, so that need to be studied further for their implementation at full pilot scale or field scale. So that's all from my side, and this is the entire setup sequence of experiments we have performed in our laboratory. And this is just one part where the dye contaminant has been utilized, has been selected as model contaminant. Currently, we are focusing over other endocrine disrupting chemicals. We are working on alkyl phenols. We are working on pharmaceutical drugs and personal care products also. So that is all from my side. Thank you so much for patiently listening.
Thank you, madam, for enlightening the nanotechnology in phytoremediation. We cannot neglect our nanotechnology because the era of nanotechnology, and we have to explore the nanoparticles for the phytoremediations. So thank you very much, madam, for sharing your views, and definitely it will be helpful for our audience, our students, to take entry in this field. Thank you very much, madam. Muskan, madam, do we have any questions from audience?
Yes, yes, sir. There's one question
Please.
from Dr. Razia. Please go ahead. That is, ma'am, can you please elaborate on the biophysical studies done to understand the reactivity of the modified nanoparticles?
Can you explain what kind of biophysical studies? I'm not getting the question actually.
Ma'am, the question is
Hello.
to elaborate on the biophysical studies done to understand the reactivity of nanoparticles.
If I'm getting this question
To understand the
correctly, so
reactivity of
if I'm getting this question correctly, the this characterization which is done for after the synthesis of nanoparticle, and then it is applied over some of the selected contaminants, say for example pesticide or any emerging contaminants you are taking in. So you have to study the effect of different reaction parameters, and this reaction parameters study can be taken up like with two approaches, one variable at a time approach, or you can also go for optimization techniques, optimization methods. Say for example, design of experiment. Currently, we are using the CCTV design for optimization of the entire reaction parameters. So that can be done through optimization, and after optimization, another next step is to check the toxicity of those nanoparticles in real environment samples. So if this the results are encouraging enough to go on, then you can subject that develop technology for practical application also. You can check it out on large scale for the textile wastewater or any pharmaceutical industry wastewater or any other industry which is generating the wastewater containing different loads of organic and inorganic contaminants. So that's how this can be done. I hope I answered your query.
Thank you, madam. Thank you very much.
Thank you so much. Thank you so much. Thank you for
thank you,
having me in
ma'am.
this platform.
Next, I would like to invite our next speaker, that is Dr. Sonia Maurya, Assistant Professor at Lovely Professional University, Punjab. And I would request Dr. Pagare to please introduce our expert. Over to you, sir.
Good afternoon, madam. I welcome you in our
Good
international
afternoon, sir.
conference that is current trends in waste treatment, reuse, and valorization. I invite you for your talk. It is over to you, madam. Thank you very much.
Okay, thank you, sir. Am I audible clearly?
Yes, madam, please.
Okay, so thank you so much, sir, for inviting me for this nice conference. And I'm very thankful to the Dr. Pankaj Chaudhary for inviting me here. Uh, shall I share my screen for starting my presentation?
Yes, ma'am, sure.
My presentation is visible to everyone.
No, ma'am, it is not yet visible.
Okay, I have shared my screen. It's not visible till now.
No, ma'am. Just try doing it again. I'll share it then.
Now it's visible.
Yes, ma'am, it is visible now.
Now it's visible, ma'am.
Yes, ma'am, it is visible. It has been shared here.
Okay, thank you so much. Sorry, I didn't get you. What you are saying?
No, ma'am, I'm saying that the screen is visible. I mean, you can start now. Thank you.
Okay, okay, thank you. So, hello everyone. Myself, Dr. Sonia Maurya. I am assistant professor in the Department of Food Technology and Nutrition in Lovely Professional University. And my topic of presentation is valorization of the food waste and its value addition. So as we know, like food is very important for everyone, and if we'll see the sustainable goal, sustainable development goal is also saying like means so much worldwidely, so much people are suffering for the food, and they are suffering from the hunger, and they are needed the food, and they are not getting food, and the reason is behind that the lost a lot of food is going to be waste. And why the food wastage is an issue, this is the reason, like why the food wastage is an issue. So food waste, when we disposed in landfills, it's going to be produce the so much harmful gases. These are greenhouse gases, which are the methane and carbon dioxide, and they can contribute to the global warming as well as the climate change. And the global warming, as we all know, like it is very dangerous for our world. And natural resources are also going to be vanished, such as the land, fresh water, fossil fuels, and human resources. So these are also wasted. So the food wastage is itself a very big issue. And if we'll talk about the water, we cannot escape the water. Like water is also very important for us, for the living purpose, for our survival purpose. So here you can see the approximately 25% of the fresh water is uh, going to be used to produce is ultimately wasted. And even as million of people are still don't have access for a good drinking water. So there is lots of problem. And if I'm talking about the India scenario on the food wastage, you can see there are a number of facts I'm going to discuss about the Indians, like where we are stand in the global scenario for the food wastage. You will see the one third of the food which going to be produced is globally for the human consumption is wasted worldwide, and it's approx 1.3 to 1.4 billion tons per year, and that is a very huge amount. And food wastage is also directly wasting of the economy. So if we'll see our India, like we Indians stands for the in the line of the food wastage, along with the China, US, and Brazil. So we are also in the major food wastage country. We are wasting the major food waste. And we are also, if we'll see the population of our we are so much in population, second in China, and second with the China. And hence we our requirement of food is also so much higher. Okay. In global hunger index in 2017, it was reported that India ranks 100 among the 119 countries in the ranking. And in India, majorly food is wasted in the different sources. If we'll see the hotels, restaurants, supermarkets, and residential blocks, cafeterias, and in airlines, and so many and so much produce is also coming. Waste produce is also coming from the food processing industry. Okay. And different industries also, industrial waste is also there. But according to the food waste index report 2021, which is given by the UNEP, that is United Nations Environment Program, India alone, if we'll see the scenario of India, from the high income group, we are wasting from the high income group 90 kg per capita per year. Food is going to be waste. And 68 kg per capita per year is wasted from the middle class family, and low income group is wasting the 63 kg per capita per year. So the range is so much higher. This is the chart which is going to give you the small discussion regarding the different sources from which we can get the different food wastes. and how we can valorize them, how we can give them, how we can do the modification and can have the value added products. So if we'll see the household food waste is also a source of the food waste. From supermarket, from hospitals, we can get the so much food waste. And this food waste we can do the some modifications into it by utilizing the techniques, by utilizing the microbial fermentation and all, and we can change them into a valued products. Like if I'm talking about the composting. Composting is the process which is very simple process. In this, the breakdown of the this one organic material is organic material into the valuable gases, uh, by
Excuse
using
me,
the
madam.
microbes as well as the insects. Am I
Excuse me,
audible?
madam.
Yes,
Yeah,
sir.
you are audible, but your slides are not moving. Yeah, it is
My
that on
slide
first
this
slide. Yeah, it is on first
just
slides only.
you are I am in first slide, but I am moving it, sir. Just a second. Shall I share my slides with you, sir?
Yeah, please, madam.
Just a second.
Yeah, it
Uh,
is moving now. Yeah, it is moving now.
no, I think I
You are in
have to
fifth slide.
share it with you, so it will be easy for me also. Like if meanwhile if it is not going to be moved, then again it will be create the problem.
Yeah,
Shall
please,
I share
please.
it with you?
Yes, please.
Okay, just a second.
Muskan, madam.
Yes, sir.
Yeah, please share madam slides. Hmm?
Okay, okay, sir.
Sir, am I audible?
Hello, madam. Did you mail the PPT?
I have mailed, but it is showing like this address is not found. And now I am sending to the Muskan Sarma. Previously, I have sent to the Rajia ma'am.
Okay. Yeah, please send it to Muskan.
Yes, sir. Only
Muskan.sharma.
give me 2 minutes.
Yes, yes.
I have sent to this PPT to the Muskan ma'am again, but ma'am is saying like I have to share my screen. So I'm again sharing my screen. My screen is visible, sir?
Not yet, madam.
Not yet. Okay, just a second. Now it's visible.
No, madam.
आप एक बार मुस्कान मैम से पूछिए क्या उनके पास उन्होंने रिसीव किया है मेरा पीपीटी.
Muskan, ma'am. Hello, Muskan, ma'am.
They are saying like address is not found. I don't know why.
Madam, it
Okay,
is
no problem.
Muskan.
It's okay. I will continue now like this only.
Yeah, please, ma'am.
So, yes, different food sources. I have already talked about like household, supermarket, supermarket food waste, hospitality sector, and composite we can prepare from these food waste so that this composite could be convert into the important biomanure and can be utilized for the crop fertilization purpose. And anaerobic digestion also we can do, and still we are doing the anaerobic digestion. It is a digestion in which we are going to utilize the organic waste, and it is going to break down to
Excuse
generate
me,
the biogases and biofertilizers. Dark
ma'am.
fermentation is also there. Dark fermentation is an alternative method for the biological hydrogen production from the biomasses. Green vehicle fuel, nowadays it is very much in matter of concern, like so many people are working on it. Like we can utilize this food waste and can utilize for the fuel purpose to gear up the vehicles. Absorbent for the pollutant removal, we can prepare so that pollution can be removed by these absorbents. So many bio absorbents are also preparing nowadays with the help of the microbes, and nanoparticles for the biomedical applications are also utilized.
Madam, your screen is visible now. Just try to move it. Huh?
Okay, now it is moving, sir. I am on next slide, that is energy recovery from food waste.
No, it is on fifth slide now. Different sources of food waste and their value addition.
Okay, now it's, sir, energy recovery from food waste.
No, no, no. I think some problem is there. Huh?
Uh,
Okay,
might be
madam.
some
Go
network
ahead.
issues are there.
Uh,
Shall I
please, please
continue,
go
sir?
ahead, madam. Yes, yes, please,
Okay,
ma'am.
okay,
Please, madam.
okay.
Hmm.
Energy recovery from the food waste, how we can recover the energy. As I told you, the composting, it is a nature's way for recycling. And this process, we can utilize the organic waste, such as the food waste and garden clipping for the biodegraded and turn it into the valuable fertilizers. Anaerobic digestions, we have already discussed about, like we can prepare the biogases, okay, by utilizing this food waste with the help of the methanogens microbes. Biofuels from the waste of the cooking oil is also there. In many places, it is a legal requirement, like the oil and the fats which we are going to be utilized for the frying purpose, they must be collected and they must be recycled so that we can prepare the biofuel for the vehicles. Here, the some indicators has been given to categorize the food waste, and this chart has been taken from the one paper which is published in 2017. Here you can see the different indicators are there. On the basis of them, we can categorize the food, like food edibility. Like is it the food waste which is edible or inedible? The state, is it a eatable or uneatable? The origin, animal based or plant based? The complexity of the product, it is comes from the single product or the mixed product? Animal product presence is there, meat product or the plant based product? Treatment, like is it processed or unprocessed? Packaging material, like the packaged product, unpackaged food waste is there. Packaging biodegradability, we can check, like biodegradable packaging is there or the non biodegradable packaging is there. Stages of the supply chain, like the waste is coming from the catering or it is coming from the non catering waste. So, what are the adverse effect of the food waste? You will see food waste are these food waste are giving the so much adverse effect to our land as well as our nature. If we'll talk about, I already discussed about the landfills. Like if this landfill emit a large amount of the gases, especially the methane, and these gases can cause the emission of the infrared radiations which heats up the environment and leads to the changes in the climate, as well as they are responsible for the global warming, and its negative effects are can be easily seen by us. Okay, our resources are going to be vanished slowly, and the large number of fresh water and groundwater is also wasted. And this fresh water, due to the wasting of the fresh water, we are not able to means most of the people are not able to get the fresh water for their consumption. And you will see, the by throwing the 1 kg of wheat, actually we are wasting the 50,000 liters of the water. And similarly, if we are wasting the 100 liters of water, when we are going to throw the 1 glass of milk in the drain, so you can see like how much water we are utilizing, how much water is required to generate the food. And that food we are when going to waste, it means simultaneously we are wasting the natural resources. Million of gallons of oil are wasted every year to produce the food, which is also wasted and goes uneaten. And agriculture chiefly is responsible for alarming threats to the plant and animal species, which are at the stake. And it was estimated by the International Union for the Conservation of Nature. Home composting diverts up to the 150 kg of food wastage per year from the local collection authorities. This is the chart is going to give you the volume of the food wasted. Like cereals is going to 30% cereals are wastage. Fish and seafoods 35% they are going to give you the wastage. Dairy products 20%, meat 20%, pulses 20%. Fruits and vegetable are more perishable actually a commodity, and they are wasted 45%, and roots and tubers are also wasted 45%. So how we can avoid the wastage, how we can reduce it? So reduce the wastage, for that we need to plan properly, like how we should going to be utilize the food, how we have to deal with the overproduction of the food, and we should avoid the overproduction of the food. Better storage conditions should be adopted in order to stretch the shelf life of the food product. The alternative market should be discovered in order to keep the food in human food chain. Food in need, surplus food should be directed to the organizations and charity which can redistribute this food. So, and this should be done in long run to avoid the alarming wastage of the food. So this we can do to avoid the food wastage. Feed livestock. Food which is unfit for the human consumption should be feed to the livestock in order to avoid the wastage. And through the restaurant and hotels, also that food which if it is good for the livestock, we should transfer it to the livestock feeding purpose. What are the impact of food loss and waste? So there are numerous bad impact on food of food loss and waste. You can see the picture. The greenhouse gases effects are obviously there. Negative environmental impacts are there. Food insecurity is going to be raises out, and reduce the nutritional status. When the person is not getting the proper food, nutritional status will be reduced, simultaneously affect the economic of the country and affect the food availability and prices also. If the food wastage will be more, then prices will be the higher, and it's also affect the availability will be the reduced. So these all are the impact of food losses and waste. So what we should do, how we can deal with this problem, the strategies for reducing the food losses and waste, we can adopt some strategies to reducing the food losses and waste. Micro level a solution we can do. So at harvesting and post harvest stage, we need to involve the more improved practices. We need to educate the farmers. We need to provide them the technical things, and provide them the new techniques. We need to aware them regarding the harvest losses, adoption of the technical innovations. We need to invest, we need to combination of these, so that we can minimize the post harvest losses and harvesting losses. And it requires the support and cooperation of the food industry retailing to the ensure that an appropriate range or portion sizes is available to meet the needs of the different households. Private investment in the production, post harvest, business, and food services could be there. Meso level solution is there. In it adapt a food chain approach that food loss wastage can be reduced through the actions. And investment in the food processing infrastructure, it invest in the adapted cold chain development, develop the food processing industry widely, so and so many industries, so that the food processing can be easily done. Ensure the proper capacity building, education, training, extension services. Unleash the crucial role of women also to reduce the food loss wastage, and allow a role for the corporate social responsibility, promote the consumer behavior changes, valorize the byproduct, side streams, and non-used foods. Macro level solutions, we'll see solutions at the micro or meso level can be enabled, supported, and enhanced by the action at macro level. And this includes the specific policies against the food loss waste, improving the infrastructures, particularly transport, energy, and market facility, and requires the government action with often involvement of the local authorities and also of the private sector. Here, some value addition, potential valorization for the some food products I have included in my PPT. You can see the apple juice process. After processing, lots of byproducts are obtained, and byproducts in the form of peel, in the form of pulp, in the form of seeds. So these byproducts can be utilized for obtaining the some value added products. Like sources, these peels are the good sources of pectin production. Pectin is required for number of preparations, food preparations such as jam, jelly, marmalade. Just a small example I am giving. Pulp and seed is the good source of quercetin, that is the antioxidant. And we can also obtain the other bioactives from the pulp and seeds. Seeds are also good source of the essential oil. So essential oil we can obtain from it. So through the food industry waste. the apple juice company waste, we can utilize these byproducts for obtaining the such products. Orange juice process. In orange juice process, byproducts like peel, it is a good source for the bakery industry ingredient. Peel is also directly utilized for making the marmalade. So peel you can use for the marmalade is the nice byproduct, means a utilization example. And in the market you can see the marmalade, jam marmalade, jelly marmalade is available. Sources of candy industry is ingredients, and it is also peel is also source for the flavor industry. You can obtain the essential oil from it, and you can utilize that oil for the flavoring industry. Many food culinary it is utilized. Broccoli, it is a frozen broccoli or canned broccoli. This is after processing, the byproducts are the stem of broccoli, and that has the high amount of vitamin C and invert sugars. Invert sugars is also having the good utilization in the fruits and vegetable industry, especially the fruit products. It is the source of invert sugar production, and it is also a source of soup production. So stem can be utilized for making the soup and sources of the garnish of the canned food and the frozen food industry. Corn, frozen and canned food. Frozen corn is also available in market, and after processing, the byproduct is corn milk and corn leaves. Corn milk and corn leaves can be utilized for making the tea, that is the herbal tea, or it's so much utilization in pharmacological products for the treatment of various disorders of the urinary tract infections and kidney. And nowadays, so much research is going on to utilization of the leaves of the corn. Okay, and tea beverages, means corn beverages are also going to be made, and it is also its cob, the cob portion is also utilized for making the chewing gum and other things, and for making the biofuels. So here I'm going to concluding my presentation in terms of economic and sustainable and social environmental consequences. Valorization of food waste and losses through the integrated biorefinery conversion to the valuable chemicals, energy, consumer goods is a feasible option for utilizing this waste. Okay, and all of this comes with a set of significant challenges that necessitate the systematic and advanced biorefinery processes design and optimization in order to ensure the chemical conservation processes and energy efficient, economically and viable, and capable of creating jobs in rural areas while having the minimal environmental impact. Thank you so much.
Thank you, madam, for focusing on food science and technology. You have done your MSc and PhD both in food science and technology.
Yes, sir.
And my student, particularly, this is my questions. Uh,
Yes.
my microbiology students always they ask, sir, after my MSc microbiology, how we can go for the food technology, because it is the very important industry and they want to enter. So kindly focus on this topic, madam, how my students will be get benefited after MSc microbiology.
Actually, basically, so they have to do the food microbiology, but microbiology students can also be go for the industry, because fermentation is the process where the so much microbiologists are required. Because if you will talk about the fermentation, this is the very huge company, huge thing. Like the student can go for the alcoholic beverage industry. They can go for the other food industry, and surely they will get the chance, because microbiologists are always important, and they are always recruited everywhere.
Hmm. Thank you, madam. Thank you for sharing
Thank you
your
so much.
views and your
Thank you
expertise.
so
Thank
much,
you
sir.
very much, madam.
Thank you, sir.
Hello, Muskan, madam.
Okay. So now I would like to invite the last speaker of this session, that is Dr. Abhay Raj. Principal Scientist at CSIR Indian Institute of Toxicology Research, Lucknow, India. And now I would request Dr. Pagare to please introduce our expert. Over to you, sir.
Good afternoon, sir. I welcome you our international conference. I'm very grateful to introduce you here. Uh, sir is Sir has done PhD in environmental science from Lucknow University, Uttar Pradesh. His major of research work during his PhD was focused on the bacterial degradation of lignin from paper mill effluents, and currently he is a senior scientist in environmental microbiology division at CSIR Indian Institute of Toxicology Research, Lucknow. His work involves development of green technology by effluent treatment for sustainable environment and health. The major areas of his research work include bioremediation, toxicity, and genotoxicity assessment, microbial biotechnology, and proteomics. He has published more than 60 research and review papers in peer-reviewed journal, SCI journal, published by Springer, Elsewhere, Wiley, and Frontiers. He has also published national and international books, chapters, magazine articles, and he's presented his research work in Indian as well as abroad also. He's also been serving as an active reviewer for various reputed national and international journals. He has successfully completed major research projects funded by DBT, Department of Biotechnology, Department of Science and Technology, and also from CSIR. He is also guiding PhD student and MSc student. He is life member of scientific organization like American Society of Microbiology, ASM, Association of Microbiologists of India, Biotech Research Society of India. Sir, I welcome you in our conference, and we are eager to listen you, sir. Over to you, sir. Thank you very much,
Thank you.
sir.
Thank you. Please share my slide from your end. Dr. Pagare, am I
Yes,
audible?
yes, sir. Yes, sir. Yes, sir.
Please share
Muskan,
my
madam,
slide.
please share slides. Just a minute, sir. We are going to share your slides. Just a minute, sir. Huh? We are going to share your slides.
Okay, sir, no issue.
I'm going to share the screen now.
Yes, is visible, but it, make it full
Yes,
screen.
sir.
Slide show mode.
Yes, sir. The slide is visible. Okay. Just let me know when you want to like change the slide. Okay. The slide
Okay.
is visible, sir.
In slide show mode, click slide show mode, madam.
Sir, actually, slideshow is not showing on the screen, so we will have to continue with the home page itself.
Home page.
Yes, sir. The slide is not showing on the slideshow.
Okay, madam, I am continuing with this mode. So,
Okay, sir. Thank you.
okay, good afternoon, all of you. I am very thankful to Sandip University for inviting me to deliver talk in this conference. So, my topic is exploring sustainable approach towards biomitigation of contaminants in paper industry effluents. During this presentation, I will show my experimental data which has done in our laboratory as well as pilot scale with collaboration of paper industry. So, Myself, Dr. Abharaj, I am a principal scientist at CSIR, Indian Institute of Toxicology. Please next slide. So this is the our institute motto. Our institute motto is safety to environment and health and service to the industry. Next slide. Our institute is mainly working in the five areas, that is system toxicology and health risk, food, drug and chemical toxicology, environmental toxicology, and nanomaterial toxicology or regulatory toxicology. I am working under environmental toxicology and dealing with the uh, industrial effluent for the development of eco-friendly technology for the treatment of industrial wastewater, including paper industry, tannery, textile, etc. Next slide. So coming to the my topics, as you know, uh, that pollutants emerging from paper industry is a global problem. At present, there is 850 paper mill in India. So India is the 15th largest paper industry in the world, and most of the paper industry in India is a small scale. It is accounting almost 76% that don't have proper treatment systems, and they have directly discharged effluent into the river system without proper treatments. Next slide. Next. This is the generalized process of paper making. Paper is made from lignocellulose biomass, that is wood, bagasse, and that is a paper. Oh, lignocellulose biomass contain lignin, hemicellulose, and cellulose. The paper is made from cellulose. To separate cellulose, there is a removal of lignin and hemicellulose. The removal of lignin and hemicellulose is performed in alkaline condition at high temperature. After pulping process, there is a separation of pulp, and the liquid is called biomass that contain lignin and hemicellulose, and the resultant pulp, that is cellulose, contain residual lignin that is a very hard to separate. And for the separation of residual lignin, there is a chlorine bleaching process, and during chlorine bleaching process, there is a formation of chlorophenols like pentachlorophenol, dichlorophenol, tetrachlorophenols, and these compounds are highly toxic to the environments and human health. Next slide. So paper industry consume large amount of water and generate almost 750 to 225 meter cubes of wastewater per ton paper produced. The effluents are generally colors, and it is highly toxic. It contains sodium salt, solvent, lignin, chlorophenols, that is measured as a OX, resin acid, tannin, sulfur compound, and these compound contributes high color, COD, BOD, TDS, and toxicity. Next slide. Next slide, please. Next slide, please. Muskan madam, next slide. Am I audible? So we have tried to develop a biological process for the treatment of paper mill effluent, especially degradation of lignin and chlorophenols. So we have our aims was to isolate, characterize, and develop a consortium of lignolytic bacteria. Lignolytic bacteria are those bacteria that are able to degrade lignin, chlorophenols, and colored effluents, including dyes. And our second objective was to lab scale treatment of paper mill effluent using a consortium of lignolytic bacteria. So I have completed these two objectives, and the research funding of the our work is published in SCI journals like Hazardous Material, Chemosphere, Frontier Microbiology, Science of the Total Environment, and RC Advances. The third objective was to optimization and validation of microbial process on a pilot scale bioreactor at paper industry site. Next slide. So in this slide, we will present a brief result of our finding. So first we characterized paper mill effluent that was collected from Star Paper Mill Saharanpur. The effluents were characterized for physicochemical parameter and toxicological parameters. So the result shows that effluent contains high BOD, COD and colors that is many times higher than the prescribed limit recommended by CPCB. The effluent contains various chlorophenol detected by GCMS, and it is a phytotoxic and cytotoxic nature, genotoxic nature. It induces chromosomal aberrations in alien SEPA and reduce plant growth. Based upon this analysis, we can say the paper mill effluent is still toxic and need proper treatment before discharge into the environment. Next slide. Next slide, please. So coming to the results, so we have isolated lignolytic bacteria from activated sludge from paper mill effluent. The sludge were enriched for the lignolytic bacteria and they were isolated on lignin agar plates. And the isolated bacteria were further tested for lignin utilization, and we found five bacteria, that is Bacillus cereus and Bacillus arbutai and Bacillus wardmani and Bacillus paramoecates and Serratia liqueficans. These five bacteria were actively involved in lignin degradations. We further tested lignin con- degradation by same analysis. In same image, you can see the control lignin powders has a smooth and flat surface, but the bacterial degraded lignin surface was eroded and rough surface. This clearly indicates degradation of lignin by our isolated bacteria. The pure culture, we are able to degrade lignin between 40 to 48%, and mixed culture of these isolates, we have degraded lignin up to 62%. Next slide. So we further tested our bacterial consortium for the degradation of pulp paper mill effluents. So after treatment of paper mill effluent with our developed bacterial consortium, there was a significant reduction of pulpants, that is a lignin, color, COD, and phenolics. The phenolics reduction was almost more than 95%. Next slide. So the removal of organic contaminants was estimated by GCMS analysis. We have detected several organic contaminants in paper mill effluents before treatment, but after our bacterial treatment, most of the compounds were diminished and new compounds were formed due to the degradation of lignin as a metabolites after bacterial treatments. This slide clearly shows removal of organic contaminants, which is a mostly persistent in paper mill effluent after secondary treatments. Next slide. Next slide. Next slide, please. Muskan madam, next slide.
Yes, sir, the slide has been changed. It is not appearing. Just wait for a minute. There was an issue. I'll share.
Am I audible?
Yes, sir, you are audible. I'm just sharing the screen.
Ah, it's here, this here.
There was just a little
Yeah.
network issue. I'm
Okay.
sharing it again, sir.
So determine the reduction of contaminants. We have performed phytotoxicity test. So it is apparent, clearly apparent in this slide that after bacterial treatment, the effluents was detoxified and it supports seed germination, which was not earlier supported for seed germination. The bacterial treated effluent also improved amylase activity and sprout length. Next slide. So, so further we have used our bacterial consortiums in laboratory scale bioreactors. This bioreactor is designed by myself. This is based upon activated cell process. There is a reaction storage, reaction, and settling tanks. So we used our bacterial treatment system process on this laboratory scale bioreactors. The flow rate of the effluent was kept 2.5 liter per hour so as to obtain 8-hour HRT. And the recirculation of active biomass from settling to reaction tank was 5 liter per hour in order to maintain active biomass in the reaction tank. So after 8-hour HRT, we found color, lignin, and phenolic reductions were 52, 58, and 85% after 12-hour HRT. With minimum inputs, we have added only 0.25% yeast extract. There was no addition of glucose and other salts. So this encouraged our results, and we further, next slide. To optimization and validation of our developed process, we have further designed a bioreactor that is 2000 liter capacity, and we have approached S paper mill near Ajodhya, near to Lucknow for validation study. We have installed our bioreactors in the industry premises, and we used our bacterial consortium for the treatment of fresh effluent that is coming from their paper making process. So on preliminary observation, we found very good result and reduction of color and lignin and phenolics were 48 and 40%, and phenolics were 65%. So in industry premises, we are optimizing our bioreactors systems in different effluent that is coming from washing, pulping, and bleaching section. The optimization and validation of process uh, at industrial site is underway. So this is the our result. We have developed technique in laboratory scale and transferred at industry scales. So this is the last slide. We are highly thankful to Department of Biotechnology, Department of Science and Technology, Department of Science and Industrial Research for financial supports. We also thankful to my research teams, and we are very thankful to Sandip University for inviting me to deliver the talk. This is the last slide. Thank you. This is a take home message: save water, save energy, and save the environment. Thank you, sir.
Thank you, sir, for highlighting the paper mill influence, which is very important, and it has to be pre-treated before the discharge in water resources. Thank you, sir, for your information and giving your valuable sharing your valuable research with us. Sir. Thank you very much. Muskan, madam, do we have any questions for sir? from audience? Yeah. Thank you, sir. Thank you very much for sharing your experience with us.
Sir,
Thank you
we
very
are
much.
also thankful to Society for Green Environment and the person of that president, Dr. Pankaj Chaudhary, is encouraged to me deliver talk in this conference.
Oh, thank you, sir. Thank you very much, sir.
Okay.
Hello, Muskan, madam.
Yes.
Yeah.
Sorry, sir, I was having some network issues. There are not any questions as of now.
Okay. Okay.
Okay, thank you, madam.
Thank you so much, sir. Thank you so much for your insightful presentation. Thank you so much, sir. Now, I would like to thank all the speakers and thank you so much, Dr. Pagare, for chairing the session. Right now, we are done with all the speakers' speeches and we will start with the poster presentations in a few minutes. The afternoon session will be handing over to Miss Vishaka Tomar ma'am in in 5 or 6 minutes. Thank you so much.
Good afternoon, everyone. I'm Ms. Vishakha Tomar on behalf of Sandip University, Nashik, the Society for the Green Environment, New Delhi, India, is blessed to welcome you all back for the afternoon session of today's e-conference on the current trends in the waste treatment, reuse, and valorization. Now, in this session, we are going to start with our oral and poster presentations. Now, there are certain instructions for the participants that you have to conclude your presentations within 2 to 3 minutes. Now, I would like to invite our first participant, Rutuja Mate.
PPT has been shared.
Yes, Rutuja, your PPT has been shared. Now you can start with your presentation.
Okay, ma'am. Good afternoon, one and all. My name is Rutuja Mate. Today I am going to present my topic on vegetable waste management. Ma'am, next slide. First, we see the introduction. In India, about 1 lakh tons of solid waste is generated every year. This means approximately 36.5 million tons annually, and from that, 94% dump on the land and 5% is compost. India is a farming country. That's why India is the second largest producer of fruit and vegetable. Ma'am, next slide. First, we see what is vegetable waste. Vegetable waste means the indigestible part that are thrown away at the different stages, such as the collection, handling, shipping, and processing. This waste includes the rotten peels, scrap portion of vegetable or slurries. This waste compressed mainly of vegetable matter, which is capable of being decomposed by the microorganism. Ma'am, next slide. Next, we see the reasons behind the vegetable waste. First is our bad weather. The weather, along with the light rains, has also hampered on the growth of the winter vegetable that are in field. Next, our processing problem. If vegetables are not processed properly, then it can cause their color, flavor, texture, and nutrition. Overproduction and unstable market. If only one type of vegetable is produced by number of farmers, then it doesn't get good market value, and because of that low value of vegetable, it includes in vegetable waste. Next, improper storage of disease. Improper storage can cause spoilage and aging, and disease of vegetable can also the reasons of the vegetable waste. Premature harvesting affects the quality of and maturity of the vegetable. Again, lack of processing facilities affects the vegetable waste. Ma'am, next slide. We will see the waste management strategy now. Do not overstore the vegetable. Buy only what you need. Vegetable waste can be treated for the biofuel production through the fermentation under the control condition. Waste management includes the collection, transportation, processing, and disposal of waste. Waste prevention, recycling, reuse, and recovery are important waste management that eases the burden of landfills, conserves the natural resources, and save the energy. Next slide, ma'am. Types of vegetable waste recycling. First is composting. It is a natural process of recycling organic matter such as the leaves of food scrap into a valuable fertilizer that can enrich soil and plant. Second is vermicomposting. Vermicompost is a product of decomposition process using a various species of worms, earthworm, white worms to create a mixture of decomposing vegetable. And biogas technology is a technique, the technique of anaerobic digestion of fruit and vegetable wastes mixed with the different waste took 55 days to produce a biogas. Ma'am, next slide. Okay, now our conclusion. Recycling and composting have grown fast in many countries. For a waste management, individual participant is also required. For a proper management, we need the governmental action quickly. And modern waste management techniques are more safe for a people and the environment than our past practices. Ma'am, next slide. And here is my references. Thank you so much.
Thank you, Rutuja. Now, our next participant is Vaishnavi, Vaishnavi Gite. Vaishnavi, sorry, there is something problem with your PPT. We'll just get you later. Now we would like to call Shrimu.
Uh, Hello,
Yes.
ma'am. Will you share my poster or will I share my poster?
Just we are sharing your poster. Just wait for a minute. We'll share. Yes, it is visible now. You can start with your presentation.
Uh, Good afternoon, everyone. I am Shrimoy, MTech Biotechnology student from NIT Rourkela, and I'll be presenting a poster on my research topic in silico analysis of bacterial enzymes for its application in dye degradation. We all know pharmaceutical molecules are extensively used globally, and the exponential increment poses growing problem of global concern. Dyes are widely used in pharmaceutical industries as coloring agents. When these dyes are discharged in water bodies, they extensively affect the biota. So, dye degradation is extremely essential. From the literature survey, it was obtained that Klebsiella species, by virtue of the beta-lactamase enzyme, are primarily primering promising dye degraders. Ma'am, can you skip down to the materials and methods, please? So, in this work, what we have done is we took the in silico analysis and the experimental analysis. And in silico analysis, first the data curation was done, and then from the literature survey, it was found out that the Klebsiella species and the beta-lactamase enzyme helps in the dye degradation. That was the protein identification. From there, we took the ligand generation. The different structures of the dyes were generated using the Marvin sketch, and then the active site prediction was done, and the docking was carried out using Autodock. Similarly, to validate the in silico analysis, we did the experimental analysis. From there, we did the media preparation using MSM, modified salt medium, and then the strain inoculation. The strain which was already pre-identified as Klebsiella species were inoculated, and the culturing was done, and then the spectroscopic analysis was done, and the result analysis were carried out. Ma'am, can you move downstairs, please? So, as you can see in this picture, from Klebsiella species, we have used the beta lactamase enzyme as our protein. PTB ID is 3C4P, and then the different dyes we have taken is sunset yellow, quinoline yellow, indigo carmine, tartrazine, and amaranth. Uh, Now moving onwards to the results. Uh, these are the different. Ma'am, can you please move up to the results? So as you can see in this results, these are the different docking structures, and basically the active site where the protein is binding with the ligand. The protein is the beta lactamase, and the ligands are the different dyes we have taken. And from there we can clearly see that the protein ligand interaction between these dyes with beta lactamase and the binding energies were recorded. The sunset yellow have the showed the maximum binding energy of minus 9.82, with followed by amaranth, while tartrazine showed a minimum energy of minus 5.8. Similarly, we carried out in experimental data, we used three strains of D7, D10, and D18 to study the dye degradation. The two parameters were considered with glucose and without glucose, and the data were taken in an interval of 24 hours, 48 hours, and 72 hours to record the dye degradation analysis. The maximum degradation was shown by was shown with the bacteria that had and the media that had glucose in it. From experimental data also, we showed that sunset yellow showed the maximum degradation, followed by carmine, amaranth, quinoline yellow, and tartrazine. So this might be one of the reasons there is a slight discrepancy between the results what we got in silico while in experimental data. In silico, this, like, although the sunset yellow showed the maximum degradation in both in silico and sunset in silico and experimental, there was some discrepancy between amaranth and indigo carmine. It might be one of the reasons because of the dye structures due to the steric hindrance or the formulation of the stable structures by resonance, which does not facilitate in the degradation as shown in silico. We will be looking into that. Ma'am, can you move downwards to conclusion? So from conclusion, we can say that sunset yellow has a higher potential of dye degradation in both in silico analysis and uh, experimental data. Our future work includes the in silico modification of the bacterial enzyme that can fasten the degradation activity, protein study for the enzyme identification responsible for degradation studies. This novel in silico approach also identifies the enzyme responsible for the dye degradation and will accelerate the screening process. Uh, these I would like to thank my college, NIT Rourkela, and as well as Sandip University for giving me this opportunity to present. And these are the references as you can see below. Thank you.
Thank you. Our next participant. Okay, so I would like to call our next participant, Ritu Mishra.
Good afternoon,
Just wait for
everyone.
a minute.
Ma'am, may I start? Good afternoon to one and okay.
Good afternoon, Ritu. We are just sharing your PPT, so you can start with your presentation. Yes, now it's visible. You can start with your presentation.
Okay, ma'am. Good afternoon to one and all present here. I, Ritu Mishra, and my friend Suraj Bidwe are here to present a topic named Bioremediation of Wastewater by Microorganism. What is bioremediation of wastewater? Bioremediation mainly uses naturally occurring microorganisms and other aspects of the natural environment to treat the wastewater of its nutrients. This type of initiative provides an avenue to environmental and economical sustainable treatment methods. Then, the microorganisms used in the bioremediation are algae, fungi, and yeast that can be used in their natural occurring forms. Then, the stages of wastewater treatment is primary treatment, secondary treatment, and tertiary treatment. Wastewater. In some applications, more advanced treatment is required, known as quaternary water treatment. This stage deals with part per million to part per billion levels of contamination and often involves oxidation of the filtration process. Each of these stages tackles different pollutants, with water becoming cleaner as it moves through the phases. Then role of microorganisms in wastewater treatment is they are workhouse of wastewater treatment system and anaerobic digesters where they are responsible for removal of pollutants and pathogens, recovery of nutrients and energy and producing clean water. Then why is wastewater treatment is important? Because wastewater treatment protects humans and ecosystem. Then the conclusion is for wastewater treatment wastewater can be discharged into the open without any legal consequences. The environment standards are very low. This leads to severe pollution of groundwater and surface water source. Due to this fact, the water that is used to supply the distribution grid is of bad quality. And I'm done, ma'am. Thank you.
Thank you. Thank you, Ritu.
Thank you, ma'am.
Now I would like to, yes, thank you. Now I would like to call Shantanu Sunavne. I would like to call Shantanu Sunavne and sharing.
Shall I share the screen or you are sharing?
Yes, yes, we are sharing the screen. There is something lagging with the screen, I think. Just wait for a second.
Okay, yes, ma'am.
Yes, Shantanu, now your screen is visible. You may start.
Yes, ma'am. Hello, everyone. This is Shantanu Sanjay Sonalne. I'm presenting my research on sludge flash brick. Next, ma'am. Uh, this is some of the content which I'm going to cover in this presentation. Uh, uh, Mainly introduction. What is brick? Brick is one of the most common masonry unit as a building material due to its properties. So the brick must be strong, economical, and nowadays it must have some environmental aspects. Looking at the surrounding. Next. This were the photos taken by us uh, in the site, uh, where it is shown that how uh, sludges are disposed or thrown away. These were some of the papers which we referred for our research. Next one. What is sludge? Uh, Sludge is basically a thick, soft waste mud or a similar viscous mixture of liquid and solid components, especially the product of an industry refining process. In this case, it is the output of a refining process of the wastewater which is led to the water treatment plant in Nashik. Next one. The another component which we used in brick was fly ash. Uh, what is fly ash? Fly ash is an effluent product by the thermal power plant which generate electricity with the help of vapors after water. As it is a waste product, we have used it in our brick. Many studies or researchers have been done from utilization the fly ash. We have used this fly ash as a bit of replacement for cement. Uh, what is cement? Uh, Cement. This is another material which we use in our brick. Cement is the most common construction material in the construction field. It works as a bonding material in the structure or in the brick when or in the brick which in which we have used it. Next now. Uh, these are some of the problem which we tried to solve. That sludge disposal is a major part of environment pollution. As in the previous slide, we saw the photos that how irrelevantly or improperly the sludge is thrown away. The product of and the second problem which we tried to solve was the procedure of making the conventional brick is quite lengthy, so we tried to make it short. Next one. These are some of the objectives which we tried to study. To perform an experimental study on a various proportion of sludge break to settle the most efficient proportion, to analyze the characteristics of a sludge break, to check suitability of application of sludge break, to provide environment friendly disposal of the waste material, to decrease the pollution with which it caused by a wrong disposal of sludge, and to reduce the manufacturing process of brick. Next one. Uh, this is the methodology which we used. The the process of making this brick consists of sieving of material, mixing all the composition, molding the brick, and curing it. Next one. Uh, the si- the first step was sieving the material. Uh, the material which we sieved was sludge. Um, the sludge was acquired from the water treatment plant, Takudi Nashik, uh, which was generated as a reduced product from the treatment plant. Uh, the same wet sludge was sun dried and used for making bricks. The sludge was sieved through the sieve size of 1.75 sieve. Then the second step was mixing. Um, we uh, we mixed the product, the materials in all the proportions which we considered, which are covered in upcoming slides. Third step was molding. This is the conventional mold which is used for making bricks, and we use the same for our sludge brick. Next. Um, the test conducted on bricks were compression test, water absorption, and drop test. Next one.
Sorry for the interruption. Can you just conclude a little faster?
Okay. The result of compression testing for proportion one, 50% cement, 40% sludge, and 10% fly ash. Uh, uh, the second proportion was 50% cement, 20% fly ash, and 30% sludge. Uh, the third proportion was 50% cement, and as respectively 25% of fly ash and sludge. The compression value of it was 15. Uh, in this ch- uh, in the previous slide, which you saw, that the most appropriate proportion as per the compression was the third proportion, 15 point, which compression was 15. Compressive strength was 15.89 Newton per annum square. Result of water absorption test. The third test was the drop test, which was taken from 1 meter, 2 meter, and 3 meter, and the brake sustained all the three tests. Future scope, changing proportion of requirement, proportion as required. Many other effluents can be used, like automobile sludge, industrial sludge, etc., and replacing of fly ash with sawdust. Conclusion: in this research, we observed that every waste material can be used for making many waste material can be used for making bricks. We also observed that the 50 25 25 proportion is efficient proportion for giving strength and absorbing less water. Sludge brick can be used in any Western river. These are the references which we used. Next one. Yeah, ma'am. Thank you.
Thank you, Shantanu. Now, I would like to call our next participant, Dhruval Pandoria.
Yes, ma'am. Ma'am, can I show my poster?
Yes, yes, we are sharing your poster. Just wait for a second. Okay, now you can start with your presentation.
Okay, ma'am. Shall I start?
Yes, yes.
Good afternoon, one and all. Respected sir, teachers, and all my dear friends. I am Dhruval Pandoria. I am from MSc in Analytical Chemistry from Sandip University. Today is my poster representation about the plastic waste management. So in my poster, what can be represent here? The plastic are very harmful to our environment and in our other living organisms. So it can be reduced. So plastic in the first above the poster, the plastic where enter in the food item is so it cause disease. With the plastic are released by the industrial waste, so it also harm to our environment in air. And so don't use the plastic or to minimize the plastic. Here it is mentioned there that don't use the single use. At the below of the poster that the plastic is also harm to the animals and birds, and it also harm to the aquatic animals, and it also harm to the ocean also. And in my other poster, it on in my other poster it's seen that the how to recycle plastic takes place. So recycling method is there. So first of all we collect the plastic material from everywhere. So it collection, and after the collection the transport the plastic in the industrial. After the transportation the sorting of the plastic takes place in the other way. After the sorting that the bundle, some plastic are recyclable and some plastic are non-recyclable. Those plastic are non-recyclable are used for making the construction of road and etc. And some plastic are used recyclable that they are using for the recyclable process. After the sorting, they are making the bundle, and bundle should be then grinding section. Then the grinding takes place into the small particles. Uh, that material are washed with some chemicals and water. And after the washing heating takes place. After the heating that form a melted structure. After the melting they form a new product in the different shape and size to form a new recyclable product. So after the recyclable product there are many recyclable products are made, like a plastic beverage water, a bottle, and rubber and container, etc. And some plastic are non-recyclable that used for the construction and road. And that's all. Thank you.
Thank you, Dhruv. Okay, now we would like to call our next participant, Harshita Arya. Good afternoon, Harshita. Now we are going to just present your poster. Just wait for a second.
Yeah, ma'am. Sure.
Yes, you can start with your presentation, Harshita.
Yes, ma'am. Very good afternoon to all. My name is Harshita Mukesh Arya. My partner name is Pratiksha Patel. And our topic name is tire waste management. Ma'am, please change the slide. Ma'am, ma'am.
पंडूरिया का हो गया ना.
Ma'am, please change the slide.
Yes, yes. I have shared. I have just done it.
Okay,
I
ma'am.
think there's some free writing. Yes.
Okay, sure, ma'am. Ma'am, slide is not visible here. Yes, ma'am. Yeah. Thank you. Recycling. Tire recycling, also known as a rubber recycling, refer to the process of recycling using the vehicle tire that cannot be no longer to use in the vehicle due to tear or repairable damage. Using that tire are among the most problematic and challenging source of solid waste. Well, this is due to the increasing number of means that the more tire getting into the landfill. And as you know already, we are talking about landfills, we are discussing and more precising issues. We are worried about environment pollution, threat of marine life, global warming, and many more issues that arise from landfill. So to avoid this, it seemed like too good idea to recycle. Ma'am, please change. You should know that every year, over 1 billion of scrap tires getting generated. In fact, in the USA alone, there is about 24.9 billion waste tires around a couple of years back. And the only way to get things done in a right way is through recycling. Ma'am, change, please. So, recycling steps are recycling steps. First, collecting of waste tire. We have to collect the waste tires. Second, tire processing. Tire processing, nothing but like converting the tire into the small pieces. Then we can handle them very easily. And at the end of this stage, we can derive the raw material that we can use as a fuel. Steel liberation. Steel gives strength to the tire. Uh, and while removing the steel and recycling them, obtained steel we can use in the formation of new tire, and the rubber and the leftover rubber we can use in the formation of rubber mulch. Screening stage is nothing but eliminating of unwanted substance and the large size of the rubber. Cleaning stage is involving in a cleaning of screened rubber uh, by using water and reducing agent. Packing and transporting the cleaning rubber. Now it's time to pack and transport to the respective industries. Ma'am, please slide change. And the byproduct obtained by the recycling of the tires are rubber mulch. Rubber mulch help in preventing soil erosion, preventing soil erosion, preventing to maintain the soil moisture. Rubber crumb help in a filling of synthetic trough. Rubber powder is used in the formation of insulators, insulating boards and sealing material. Tire drive aggregator used in a building material. Tire drive fuel is used as a fuel in the industry. Due to the some applications is not able to use in a daily life home or in a normal normal life. Steel is used in a manufacturing of new tires. Black carbon is used in the formation of insulating electric equipments. Ma'am, please slide change. Tire pyrolysis. The waste tire is doing by waste tire. Now we're having pyrolysis process. Thermal degradation of the organic component of the tire at 500 degrees Celsius to produce oil, gas, char, and wires. The oil is then CN. Okay, ma'am. Okay, ma'am. Change. Carbon black. Carbon black is converted into cable, shoe rubber, and batteries, automatic interior, ink, paint, and etc. Ma'am, change please. The innovating ways using the tire waste are this. Please change. Waste waste tires are dangerous as they are beneficial because they are non-biodegradable and energy efficient. They fill up the land space, releasing the harmful gases, causing the city's environment evoke. At the same time, when we recycle the tire, we create the new product, providing employment and revenue for the government. Providing this prevent diseases and environment pollutant and eliminating other dangerous. Change, please. Thank you. Thank you, Sandip University, for giving me such a great opportunity to represent my project. Thank you. Ma'am, I'm done with that. Ma'am? Hello.
Yes, thank you, Anshika.
Thank you, ma'am. Thank you.
Now, I would like to call our next participant, Patience Vanda. Next participant, Anshu Priya Kumar.
Yes, ma'am.
Good afternoon. I'm just sharing the PPT. Just wait for a second. Yes. You can start with your presentation, Anshu Priya.
Good afternoon, everyone. My name is Anshupriya Kumar, and my partner's name is Vishakha Paul. And today I present to you is the poster which we made for water reuse and water management. What wastewater is any water that requires cleaning after it is used. This includes water that has been used for laundry, bathing, dishwashing, toilet garbage disposal, and also industrial purposes. Water waste also includes rainwater that has accumulated pollutants and it's rain into oceans, lakes, and rivers. Pollutants are unwanted chemicals or materials that contaminate air, soil, and water. The goal of wastewater management is to clean and protect water. This means that water must be clean enough so that it can be used by people for drinking and washing, and by industry for commercial purposes. It must be clean enough to release it ocean into oceans, lakes, and river after it has been used. Wastewater is usually divided into two major groups: point source wastewater and non-point source wastewater. Point source wastewater includes wastewater that enter waterways such as lakes, rivers, and oceans from defined locations. The most common point sources are sanitary sewage and storm drains. Non-point sources wastage is water that is non-connected to specific sources. This includes runoff water that drains away from the agriculture and urban city areas, and acidic water from mines. In many ways, point source wastewater is much easier to manage because it is source and because of its source and the pollutants it contains are known. Non-point source wastewater, on the other hand, is both hard to identify and treat. It is said that less than 2% of Earth's water supply is fresh water, or of all the Earth's water, 97% is salt water found in oceans and seas. Only 1% of Earth's water is available for drinking water, 2% is frozen, and hence we should consider ourselves to reuse the water and manage the wastage that comes with it. Thank you. Ma'am, I'm done.
Thank you, Anshu Priya. Now, I would like to call our next participant, Klenia Dubai. Good afternoon, Klenius. I'm just sharing your PPT.
Yes, ma'am.
Yes. Just wait for a second. I'm sharing your PPT.
screen में
Yes, Glenn, yes, you can start with your presentation.
um, my name is Clenas Due. I'll be presenting with Patience Panda. We are in the second year microbiology, BSc, School of Science. Our presentation is about artificial intelligence in waste management. Introduction. Artificial intelligence is the ability of a computer or a robot controlled by a computer to do tasks that are usually done by humans because they require main intelligence discernment. Artificial intelligence in robotics are constantly improving the process we rely on to collect, transport, sort, and process all types of waste from medical waste to biohazard waste. Intelligence beams. These are beams with sensors that helps the waste waste companies uh, so that it allows the frequency of carrying and collecting the waste. Next, ma'am. Computer vision, automation, and machine learning algorithms allow these sensors to distinguish between different types of baggages as the baggage are being filled. For example, an intelligent waste being developed by Bin E uses machine learning to identify and categorize and sort waste immediately after it's thrown away. Smart bins are often paired with an app that lets users know the location of the nearest available bins, preventing from overflowing. The benefits of smart bins. Smart bins saves your cleaning time. Smart bins reduce the wastage of plastics bags by up to 90%. Smart bins helps your cleaning team makes more efficient schedules. Smart bins helps you plan your space better. Smart bins notifies you before your bin overflows. Flash bins helps analyze environmental awareness campaign. So with the use of the application that is in these phones nowadays or computers, they can help the people to see where the bins are located so that they can go to the bin that is aimed and put the waste. Next. So introduction of artificial bins and the uses of robots in sorting could in the future make a significant contribution in the production of high quality secondary raw materials from waste to the sense of recycling economy within the time framework of a pilot study financed by Europe Institute of Innovation and Technology, Climate KIC, a European research consortium led by Ferrovial um, Service International and uh, with Wuppertal Institute tested and evaluated such waste sorting plant and tested on real municipal waste. The result has been evaluated with regard to sort quality and purity of the relevant material flows. Next. So the importance of artificial intelligence bins. It helps in waste separation. They have numerous sensors such as computer vision which enable them to monitor the waste stream. The artificial intelligence powered robots identify the waste material and the robots arm gets hold of them. This machine can sort rubbish under various categories and even determine the appropriate method for disposal. This is uncommon for people. This is intelligent trash cans. This is uncommon for people to litter around trash cans. They are always in the hurry, which is understandable, as they live in a fast-paced world. If people miss a shoot, they do not have to time to go and pick up the baggage, so they have to go on their way. This is good thing. Intelligence trash cans, because they've got sensors that robots with arms, it picks the waste that is left when the person misses the bin. Next. Another important of artificial intelligence in waste, localizing greenhouse gas emission at landfills. Greenhouse production. What is worry is what is worrying is that greenhouse gases contribute to the warming of Earth. This in turn brings about changes to the normal weather. So the United States have the highest total quality of methane generated by humans. This does not really come as a surprise considering that need for electricity production. The good news that computer scientists have invented mobile robots which are capable of localizing greenhouse emission. They are invaluable tools for solving problems such as gas leak and explosion. European Union authorities have shown interest in gas sensitive robots and for good emission reason too. So these are the examples of the artificial intelligence beings. They are smart because when a person leaves the litter, I'm not explaining. When the person misses the bin, the bin have got sensors on it that it will check the litter or the baggage and put inside the bin. And also it is computerized with a robot, also robotic, that if it is full, it will also compress on its own. And also if it is full, it have got a signal, it will give signals to the computer, to the municipality, so that they will go and check and empty the bins. Thank you. Thank you. Any question, ma'am?
Thank you, Phineas.
Any question, ma'am?
No, there is no question from anyone.
Okay, ma'am.
Yes, thank you. Now, I would like to call our next participant, Prakash Chitodia.
Good afternoon, ma'am.
Good afternoon. Just wait for a second. I'm sharing the PPT.
Yes, ma'am. Ma'am.
Yes, Prakash. Just for a minute. I'm sharing. Yes. you may start with your presentation.
Good afternoon to all of you. My name is Prakash Chitodia, and my partner name is Kalpesh Gosavi. We are student of third year BSc Microbiology, and our topic name is an introduction to waste water treatment technology. Ma'am, please change the slide. Wastewater treatment is a process which use contaminate from wastewater convert into influent that can be returned to water cycle. Once returned to the water cycle, the influent creates a acceptable impact on the environment or various purposes called water. The treatment process takes place in wastewater treatment plant. There are several kinds of wastewater which affects the appropriate types of wastewater treatment plants. For industrial wastewater treatment, either takes place in separate industry wastewater treatment plants or in a sewage treatment plants, usually after some form pre-treatment. Further types of wastewater treatment plants includes wastewater plants, leach treatment plants. Ma'am, change the slide. Types of wastewater treatment technologies: sewage treatment plants, influent treatment plants, activate sludge plants, common and combination influent treatment plants. Here I would like to give some information about them. Sewage treatment plants is a type of wastewater treatment which aims to improve contaminant from sewage to produce an influent that is suitable for discharge to the surrounding environment. Second, effluent treatment plants, commonly known as ETP, and it's used for treating the wastewater to meet pollution code. Let dispose the treatment contaminant in form of organic matter, heavy metals, suspended solids. Third, the active sludge process is this treatment process. In this treatment process, air and oxygen is blown into raw sustainable sludge to smash the solids and develop an biological soup, which digests the organic content and pollute in the sewage. Fourth, common influent treatment, CETP. Plant's concept is evolved to effluent collect treatment effluent from various industry at the centralized facility to avail benefit and scale of operations such as plants are known as CETP, combined infinite plant. May change the slide. Chemical used in wastewater treatment: aluminum sulfate, sodium aluminate, polymer, sodium hydride, polymer chloride. Name change the slide. Wastewater management by different countries. In India, wastewater is currently being used for irrigation, gardening, flushing, cooling, or air conditioned system as feed for boilers and as processed water of industries. In China, national policy has been developed that promotes the development of water efficient technologies and encourage the issue of ready-made municipal water in agriculture first and then for industry and municipal uses. In Japan, reclaimed wastewater is used for toilet flushing, industry, stream industry, and flow arrangement to create urban image such as green space. Ma'am, change the slide. Conclusion. This present examined arrangement issue and technological options relate scale of collection and treatment systems. There is increasing movement opening behind the nation and the recycling loops inform the generation to the point of treatment and reuse must be shorter. This leads to several pollution of groundwater and surface water source to the water that is used to supply the distribution guideline of bad quality with all the pollution present. Here is a picture which shows all this. Ma'am, please change the slide. Ma'am, I have taken reference from wastewater engineering treatment disposal metallic ADDI 1979, fundamental of wastewater treatment and engineering human referred 2012, wastewater treatment plant design E Amulam 2003, and websites which are shown. Change the slide now.
Yes, Prakash, thank you for your presentation. Okay, now I would like to call our next participant, Pooja Kawale.
Hello, ma'am.
Hello, good afternoon. I will just sharing.
Good. Yes, good afternoon, ma'am.
Good afternoon. I'm just sharing your PPT. Just wait for a second. Yes, Pooja, you may start with your presentation.
Okay, ma'am. Good afternoon, respected sir and ma'am. My name is Pooja Kawre from Analytical Chemistry. Today's poster presentation topic is wastewater treatment, recycle, reuse, and valorization. The main goal of the wastewater treatment to allow human and municipal effluent to be disposal of without danger to human health or unacceptable damage of the natural environment. There are four top types of wastewater treatment plant. First one is sewage treatment plant, industrial wastewater treatment plant, agricultural wastewater treatment plant, litter treatment plant. For today's presentation, we are seeing topic which is sewage treatment plant. Sewage treatment is a type of wastewater treatment which aim to remove contaminant from sewage to produce an effluent that is suitable for discharge to the surrounding environment or an intended reuse application, thereby preventing water pollution from raw sewage discharge. Now we see the water recycling process. Now we are water recycling, so there is process of recycling. First one is wastewater contain high concentrated pathogen such as virus, bacteria, also having garbage. So into first process for raw sewage bar screening. Its process mechanical filter used to remove large objects such as rags and plastic from wastewater. Then the grit chamber removal of inorganic material such as stones, sand, also soil particle are removed from water. There is primary settling is the remove suspended solid and floating organic material. Then operation in an activated sludge process is based on pumping air into a tank which promote the microbial growth in the wastewater. Then secondary settling. In secondary treatment is the removal of biodegradable organic matter from sewage or similar kind of wastewater. Then sand filter, the water to be purified slowly sink through the sand of the sand filter, leaving the dirt particle from the water behind in the fine core of the sand, in which harmful bacteria is killed by the process of disinfection, and finally over raw sewage water ready to outfall or reuse. What water can we reuse? Rainwater, stormwater, grey water, wastewater, and industrial, commercial. Then wastewater treatment and reuse is a good way to meet large part of your water needs. Wastewater in this context refers only to grey water, that is water from bathroom, sinks, washing machine, etc. Water from toilet and kitchen, which makes it easier to treat and reuse than black water. The sewage water coming out from house is not suitable for drinking. The water should be further treated using method like ozonation to make it portable. And this recycled water we can use for agriculture, landscape, and also industrial purpose. And some of water we can again treated in advanced purification facility. And this purified water stored in reservoir and then one again purify this water with the help of drinking water treatment, and then we can use that water in the house for drinking and cooking and any other purposes. Thank you, ma'am.
Thank you, Pooja. Now, I would like to call our next participant, Vaishnavi Geete. Good
Yes,
afternoon,
ma'am.
Vaishnavi.
Good
Yes, I'm
afternoon.
just okay. I will just share your PPT. Just wait for a second.
Shall I start, ma'am?
Yes, Vaishnavi, you may start with your PPT.
Myself, Vaishnavi Gite from Sandip University. So let's begin. Topic for my presentation is e-waste. Let's see it. Next slide, ma'am. Next slide, ma'am. So let's see first what is mean by e-waste. According to the e-waste management rule 2060, e-waste means any old electrical and electronic equipment whose life has been ended or in part discarded as waste by the consumer or bulk consumer, as well as reject from the manufacturing industry. And second is what is mean by e-waste recycling. It is the reuse and reprocessing of electrical and electronic equipment of any type that has been rejected or regarded as end of life product.
It includes home equipment, ICT equipment, and other electronic utilities. Next slide, ma'am. E-waste recycling plant is a facility where end-of-life electronic product is transported for recycling purpose. The e-waste collected from the consumer is transported to the e-waste recycling plant for segregation, dismantling, treatment, and disposal. Next slide. Ma'am, next slide.
Yes, Vaishnavi, it's just lagging, and I will share it again.
Okay, ma'am.
It's Vaishnavi. There is some technical issue. We are looking for your file. Just wait for a second.
Okay.
Sorry for the inconvenience. You may start with your PPT.
Okay, ma'am.
Yes.
So let's see. First, what is mean by e-waste? According to the e-waste management rule 2060. Okay. This then certain examples of sources of e-waste and their related adverse impact are shown in the following table. Next, ma'am. These are the quantification of some of the toxic elements which are present in average computer weighting, which is approximately 31% 0.5 kg. A typical computer monitor, it may contain more than 6% lead by weight, much of which is in the lead glass of CRT. Up to 38 separate chemical elements are incorporated into e-waste 18s. Next slide, ma'am. Process of e-waste. Then we collect e-waste, save storage, then it automatically separate. Next slide, ma'am. Response e-waste processing technology. It consists of cutting mills for electronic circuit board, wire, and small part, granulation mill for e-waste with primary separation, vibratory feeder assembly for granulation mill, enhanced air purification system, convertery bed, it is of 10 feet, magnetic separator, it separate ferrous component. The non-ferrous ferrous fraction is passed over a convertery belt to the granulation mill by a vibratory belt. The fractional separator separate the granulated material into non-metallic and non-ferrous mixed metal. The entire process is completely mechanical. No chemicals are used at any stage. The entire process is free from any kind of polluting process. Next slide. According to forecast, based on logical model and material flow analysis, the volume of obsolete pieces generated in developing region will exceed that of developed region by 2060 to 2080. By 2030, there would be two obsolete pieces in the developing world for every obsolete piece in the developed world. Similar forecast has been arrived independently. Next slide, ma'am. The recovery method, it is followed by the unit in the unorganized sector in India for various components that are described in the table. The recovery of the computer from e-waste depend on their market value, while the residue and the leftovers such as ashes and plastic residue from IC disposed of in landfills. Next slide, ma'am. This data shared of industrial operation, it indicate 1 ton of computer. It can be recover 20 kg of ferrous and 29 kg of non-ferrous. It is given from e-Parisara. E-Parisara, it is an eco-friendly e-waste recycling unit on the outskirts of Bengaluru. It has the capacity of recycle 3 ton of e-waste every day and is expected to be scaled up to achieve 10-ton capacity in 5 years. Next slide, ma'am. It is the material flow of brand new and second hand television set among Japan, China, and Hong Kong. Next slide, ma'am. Some advantages and disadvantages. Advantages are it reduce greenhouse gas emission, reduce pollution, it protect biodiversity, conserve natural resources. Some disadvantages, cost is high, so then it is impossible to recycle 100%. Next slide. Thank you, Sandip University, for giving me this opportunity. Thank you very much. Ma'am, it's done.
Thank you, Vaishnavi. Now, I would like to call our next participant, Ria Nandi. Good afternoon, Ria. I'm just sharing your
Good
PPT.
afternoon, ma'am.
Just wait for a minute.
Yes, ma'am.
Yes, Ria, you may start with your presentation.
Okay. Good afternoon, teachers, respected teachers, friends, and all my dear friends on the who are watching this video. Thank you, and be patient and please watch the video. Ma'am, first slide. Today my topic is about bioremediation. Bioremediation is one of the core topic which we use basically to treat the waste, and I will talk about it in a little abstract only. First of all, bioremediation is first of all we can see that where bio means living organisms and remediation is a cure. So in total, the definition of bioremediation is that is a process that use microorganisms, plants, or microbial or plant enzymes to detoxify the contaminants or the pollutants. There are many type of pollutants, as we can see: heavy metals, radioactive pollutants, pesticides, explosive fuels, etc. And bioremediation, if it is done by bacteria, If the bioremediation process is done by bacteria, then it is called as bioremediation. If the living organisms which we use to detoxify the pollutant, then if we use as fungi, then we can call it as mycoremediation. And if plant is used, then phytoremediation, and if algae is used, then phycoremediation. These four categories are basically in bioremediation. Now we will start with bioremediation. There are two types of bioremediation. First, ma'am, second slide. First, we will see about in situ bioremediation and ex situ bioremediation. In situ means that treatment at the site. It means the pollutant where it is present. We are treating the pollutant at that place only. We are not taking out from there and placing it somewhere else, because it will contain some cost and it is expensive. So in situ we basically do the treatment at that place where the pollutant is present, and as it is we don't have to, there is no transportation cost, so it is less expensive. And in ex situ, what we do is we take away the pollutant at some place, and at that place we degrade it, and it is more expensive, as if it is a water we use pump. So for pump high energy is invested, and this is more expensive. Excavation and pool of water is there. Ma'am, third slide. Now, in situ means in situ bioremediation means there is no need to excavate or remove soils or water in order to accomplish remediation. It basically means, now some types of it are bioaccumulation. Bioaccumulation basically it means that a bacteria is present at that place. The bacteria uptakes the metal or the pollutant inside itself and stores it, and after some time it detoxifies it. It is called as bioaccumulation. After that, we can see biomineralization. Biomineralization basically means that when a soluble form of pollutant is present and it mixes with any complex molecule, like metal is mixed with H2S, hydrogen sulfide, then we get metal sulfides, and metal sulfide is insoluble. So we can basically see that this is biomineralization, and soluble form of metal is converted into insoluble form of metal. Now we can again, the second is biotransformation. Biotransformation is that the metal is present in oxidized form. Now bacteria produces some enzyme to treat the oxidized form and make it in the reduced form. This can be called that with the help of enzymes, then we can see that it is called as biotransformation. And after that we can see metal plus organic acid plus soluble metal and OH. This can be called as bioleaching. Bioleaching is about that when a metal is present and it is mixed with some organic acids produced by the bacteria, and it forms a soluble salt, and bioleaching means it moves underground after some time. The metal, soluble metal and salt is it moves down the, it will move into the below layer. Then we can see precipitation also. Biosorption is also present, that the metal binds to a metal binding protein present on the bacteria. It can be called as biosorption. Ma'am, next slide please. Ma'am, after that slide. Here we can see that bioaccumulation, some diagrams are given of bioaccumulation and biomineralization. Biotransformation and biosorption I have already told. Biosorption is a metabolically passive process made primarily by non-living organisms or biological materials, example agriculture waste. Biosorption is an efficient, selective, fast and economical process that makes the technique favorable for bioremediation of glyphosate in an aqueous environment. Glyphosate can be, as we know, glyphosate is one of the type of herbicides. Now we will speak about bioaugmentation and biosorption. Bioaugmentation is a process in which the bacteria is indigenous bacteria and exogenous bacteria. These two types of bacteria are present when microbial remediation we talk about. Indigenous bacteria are those bacteria which are present, which we naturally found at that place where the contaminant is present. We don't have to we don't have to give bacteria from outside. The bacteria is itself present at that point. So it is basically easy to handle because they are adapted to that situation. So in bioaugmentation we do this, that culture of indigenous species in a lab, and then we send then we expose it to polluted side, and the rate of degradation will increase as we multiply the indigenous species into a large number. And biostimulation is that in this we add fertilizer or nutrients which will act as an oxidant or electron donor for the species, and these are also done by the indigenous microbes to treat the polluted site. Ma'am, next slide. Ma'am, next slide. And these are the basic differences about bioaugmentation and biostimulations. First we will talk about the definition which is present here. Bioaugmentation is the process of adding specific microorganisms to enhance the existing populations and promote the biodegradation process, while biostimulation is the process of adding electron acceptors or electron donors or nutrients to stimulate naturally occurring microbial population in the contaminated area. Now about utilizing organisms are bioaugmentation is exogenous microorganisms. Why we have called as exogenous microorganisms? Because the indigenous species are cultured in the lab, so it can be called as exogenous microorganisms. And biostimulation, and they are indigenous microorganisms. Addition is cultured microorganisms, and here nutrient and electron acceptors are mainly added. We have talked about bioventing. Bioventing is a type of in situ bioremediation technique that stimulates the aerobic degradation process. It enhances the intrinsic capability of indigenous microflora to degrade the organic contaminants absorbed to soil by introducing oxygen into the unsaturated zone. Means that the pollutant may be very below the soil. Then what we have to do, that we have to provide aeration underground also. We introduce some pumps and some some tubes, so that the oxygen which is provided to basically to the bacteria for degradation of the process, so that oxygen should go inside also, so that the bacteria can depollute that portion also which is not available above. Now we will talk about ex situ bioremediation. Ma'am, next page.
So, sorry for interruption, Ria. Just can you just go a little faster?
Yes, ma'am. In ex situ we have three types: composting, land farming, and biopiling. In composting is materials like straw, bark, and wood chips are mixed with contaminated soil, piled into heap to maintain the air and air holding capacity. In composting, there are three types of composting: static pile composting, in vessel composting, and windrow composting. Land farming, a lined bed, the soil is usually spread so that it is about 8 inches thick all around. Land farming is the most simple or three types of solid phase, and it involves mainly used for non-volatile and organic compounds. And ma'am, let's start about biopiling. Biopiling is basically piling of contaminated soil within a lined area to prevent leaching. The piles are covered with polythene and liquid, nutrients applied to the surface. Now we will talk about slurry phase. Basically, triphasic system is present in slurry phase: water, suspended particulate matter, air. And now we will talk about, and there are two types of slurry phase: aerated lagoons. Treatment of municipal wastewater is basically done by aerated lagoons. And lower shift shear airlift reactors. Lower shift airlift reactors is used when waste contains volatile compounds. Now there are some examples of some microbes and pollutants in the following slides. And lastly, we will see about that why it is better that bioremediation is done by oxygen, in presence of oxygen, because the product after which we get the product is carbon dioxide and H2 water. But if it is done in an anaerobic process, then maybe it may cause it sometimes produce carbon dioxide, plus we can get some intermediate materials which can be less toxic, more toxic, or equally toxic. And And summary is given. Bioremediation is a waste management technique which involves the use of biological microorganisms to remove or neutralize pollutants from the contaminated site. Bioremediation is a treatment that uses naturally occurring organisms to break down hazardous substance into less toxic to non-toxic. Technologically, generally classified as in situ and ex situ bioremediation. And ex situ involves removal of contaminated material to be treated elsewhere. Ma'am, last slide. And ma'am, thank you, ma'am.
Thank you, Ria. Now, I would like to call our next participant, Vaishnavi Mandole. Good afternoon,
A
Vaishnavi.
very good afternoon. Good afternoon, ma'am. A very good afternoon to everyone. Myself Vaishnavi Niti Mandole and my mate is Tejasvi Kade Aire. Today I'm gonna represent my PPT upon current trends of physiochemical methods for remediation, in which I'm gonna discuss about the waste to energy. How can we convert waste to energy? Ma'am, can I have the PPT?
Yes, yes, yes. I will share my PPT. Yes, Vaishnavi, you may start with the presentation.
Okay, ma'am. The
Yes,
name
ma'am, please
itself
continue.
defines that what is waste to energy, how we convert our daily kachras or whatever the thrush come from our household or any agriculture or any kind of thrush, how can we convert that into energy? And we're gonna discuss detailed about the plasma gasification. So the current situation of our country is that about 0.1 million tons of municipal solid waste is generated in India every day, and the per capita of every each person every day is about 0.26 kgs, and about 94% is dumped on land, only 5% is composted, we call which we are done by the recycling animal feeds, biological representation, incineration. And all this landfill which contains the thrush contains the fossil fuels, which is very hazardous to the land and to us too. Man, can I have the next slide? And the future possibilities is going to be more worse because plastic is gonna rise from 4 to 6%, metal is gonna rise from 1 to 4%. Everything is gonna take a high peak uh, in the thrust management. So, what is the major thing we can do and how we convert this waste to energy is the major need for our country. Waste to energy technology converts waste into various form of fuels and can be used to supply energy. And waste feedstocks can include municipal waste, MSW, construction and demolition debris, agriculture waste such as crop silage and livestock manure, industrial waste, coal mining, lumber mills, and other facilities. And what are the advantage of waste to energy? It's like एक तीर पे दो निशान है, जहाँ से लैंड भी कम यूटिलाइज होगी और जो इन्वायरमेंटल इश्यूज है जिससे फॉसिल फ्यूल क्रिएट्स होते हैं वो भी कम हो जाएंगे. And about the gasification, and one is another method is incineration. Incineration, it consists combustion which requires the heat, and it creates many hazardous chemical or harmful gases, which is very harmful to the environment and to us too. But in gasification, there is no combustion and there is no slag at all. जहाँ पे तार क्रिएट ही नहीं होता है, जिससे कोई उसका वेस्ट नहीं निकलता, इवन द एशेस आर नॉट इवन क्रिएटेड. So it's the best thing that we can bring gasification in our country, or we can use, or we can plant a plant. So जिससे कचरा भी ना हो और उससे बहुत सारे हमें एनर्जी भी जनरेट करके मिले. In gasification, mostly methanol, ethanol, propanol, and other biodiesels are always also created. But the only disadvantage of this is कि उसके जो बायो प्रोडक्ट्स होते हैं वो बहुत कम अमाउंट में जनरेट होते हैं. And what is plasma gasification? We all know that plasma refers to the fourth state of matter, solid, liquid, gas, and the fourth one comes as a plasma. Ma'am, can I have the next slide? This is a plasma torch where the plasma torch is given and the ma'am, can I have the next slide? Next, ma'am. So this is the plasma gasifier जहाँ से ऊपर से कचरा आता है, नीचे फ्यूल और वेस्ट इन होता है, गैस आउट होता है और मोलिटन सिलिकेट और मेटल आउट इन द सीक्वेंस. मैम, कैन आई हैव द next slide? So what happened in this process? The superheated blast of air, which is enriched with the oxygen, is provided at the bottom of the gasifier, and the stereometric amount required for gasification. A very specific amount of oxygen is required, and the plant contains about 100 degrees Celsius temperature. And even it has the outer layer of jacket, which contains the water for the cooling and maintaining the temperature of this plant. So due to which जो वो वाटर सप्लाई होता है उसके अंदर, जैसे ही वो आउटलेट होता है तो वो हीट जनरेट करता है और उससे थर्मल प्लांट में यूज होके इलेक्ट्रिसिटी जनरेट होती है. And it has also has the lower melting zone. Ma'am, can I have the next slide? Yes. So advantage of the plasma gasification is it is greater feed flexibility, including coal mines, mining waste, lignite. Even अगर वो 1000 डिग्री सेल्सियस के ऊपर भी कोई अगर हमारा मेटल या फिर कोई भी है जिसको हाई बॉइलिंग पॉइंट लगता है मेल्ट होने के लिए, तो भी वो जो वेस्ट होता है, वो जो स्लैग होता है, वो भी वेस्ट नहीं होता है. It is also used in the construction site. According to the WPC estimation, only 2 to 5% of total energy input into their gasification system is consumed by the plasma torch, and that 80% of total energy input in the feed can be recovered in the produce syngas. Syngas मतलब synthetic gas जिसमें biodiesels भी include होते हैं, जिसपे हम airplanes and other जो vehicles होते हैं वो भी हम रन कर सकते हैं. And the concern regarding plasma technology has been drawn some questions from environment because it contains could left in the byproducts, but it is very in a few amount, a very few amount जो हम use कर सकते हैं in the construction sites or other sites. And only the disadvantage of this plant is that we need a land to plant this plant, and it requires a huge amount of cost for investing in it. और जो हाई फीड स्टॉक रिजल्ट्स में जो आता है, जो सिंथेटिक गैस का प्रोडक्शन होता है वो थोड़ा कम होता है, सो उसका जो रिवेन्यू है वो कम हो सकता है. बट इट्स अ मेजर नीड फॉर अ कंट्री टू लुक इनटू दिस. एंड हमारे कंट्री में जो भी जितना भी लैंड कवर होता है तरह से और जिससे भी उससे हार्मफुल फॉसिल फ्यूल्स निकलते हैं वो शायद कम हो जाए, एंड इट कैन कंट्रोल द पोल्यूशन टू. मैम, नेक्स्ट स्लाइड. थैंक यू सो मच.
Thank you. Ms. Jyoti to present.
Yes, ma'am.
Yes, I will just share the PPT. Just wait for a minute.
Okay, ma'am. Okay.
Yes, you may start with your presentation.
Okay. Good afternoon, all of you. Today I am presenting antibiotic resistant bacteria emerging pollutant from pharmaceutical industrial waste. Next slide, ma'am. The pharmaceutical, excuse me, the pharmaceutical The pharmaceutical wastewater has a mix of large concentration of antibiotic and antibiotic resistant gene, and these sites act as a hotspot for environmental contamination and the spread of antibiotic resistant bacteria. Pharmaceutical antibiotic production in large scale and their waste treated by the effluent treatment plant. The antibiotic wastewater contain the many pollutant like organic and inorganic pollutant, and they affect the environmental reservoir like human, animal, plant, and aquatic environment, and contain the risk of environment. Ma'am, next slide, please. Ma'am, next slide. I performed the resistant profiling test. Antibiotic resistant bacteria isolate from the pharmaceutical wastewater against the use 13 antibiotic and the standard disk diffusion method. The strain were overnight grew in Luria bitrani medium at 30 degrees centigrade for 24 hours. Then the bacterial suspension were adjusted to OD 600 nanometer as 0.5 100 microliter was spread on the Muller-Hinton agar plate as standard use 0.5 McFarland standard. The antibiotic discs were put on these freshly prepared bacterial known on Muller-Hinton agar plates and incubated at 30 degrees centigrade for 24 hour and 48 hour. And the degree of resistance comparing with the standard antibiotic disc sensitivity testing method. Uh, this method standard use as UCOST. Next, ma'am. And the results show the this image antibiotic sensitivity at resistant profiling of isolated bacteria uh, in this study use the I found the say, 14 isolated bacteria on resistant mostly the present resistant, and these images show the mostly bacteria are resistant or zone inhibition measured by the millimeter or the 13 antibiotic used in this study: ampicillin, erythromycin, kanamycin, tetracycline, gentamicin, ciprofloxacin, vancomycin, nalidixic acid, azithromycin, rifampicin, and penicillin G. Next, ma'am. Next slide, ma'am. And this table show the resistant percentage and MR index. MR index value represent the 0.02. This mean is the resistant value above the
these and the graphical representation of the resistant bacteria. And ma'am, next slide. Ma'am, next slide. Ma'am, next. Ma'am, next slide, please. Hello.
Yes, ma'am. Yes. I've changed it, but I think it's log. Like, just a second.
Uh, I identify the five potential bacteria according to the disk diffusion method, and I test the morphological and biochemical test. As you can see that I perform the biochemical test: indole, motility, citrate catalase, MRBP, and starch hydrolysis. Ma'am, next. The present study was determine the multiple antibiotic resistant among the bacterial strain that were isolated and identified from the pharmaceutical wastewater. In conclusion, most pharmaceutical water, treated and untreated water isolate, were found that uh, the multiple MR index and five potential bacteria 100% so resistant to the 13 antibiotic. They could be responsible for the dissemination of the antibiotics resistant in the environment and also of many other environmental risks. The isolated multi-resistant bacteria pose high risk in the human, animal, and environmental health. In other hand, these antibiotic-resistant bacteria could be a potential agent for bioremediation of antibiotic pollutant environment, and especially that strain can use the antibiotic carbon and energy source. Future study of molecular identification and develop consortia of these potential antibiotic-resistant strain. Next, ma'am. Thank you, ma'am. Thank you.
Thank you. Okay, I would like to call our next presenter, Suraj Suryavanshi.
Yes, ma'am.
Yes, good afternoon. I will just share your PPT. Just
Am I
let me
audible,
a
ma'am?
minute. Yes, you are audible. Yes, Suraj, you may start with your presentation.
Okay, yes, ma'am. Okay, yeah. Um, Hello, everyone, and good afternoon, respected all teachers. Myself, Suraj Balaji Suryanshi, student of MSc Life Science, Sandip University, and I am going to present here the topic is fabric waste and their impact on environment and also on human health too. Next slide, ma'am. Next slide, please. Okay. Here is this is the vast concept, but our daily using fabric uh, have toxic impacts on air, soil, and water too. Here is one quote of Mahatma Gandhi: "There is no beauty in the finest cloth. if it make hunger and unhappiness. Okay, so our growing fast fashion, this is the monster in our closet. It has become challenge to wear a garment more than 5 times. Why? So, garments quality is declining every year. As a result, our clothes immediately look faded, shapeless, or worn out. Trends are changing so quickly that we cannot keep. We continue to purchase just to stay to date. Okay. This fast fashion mass production of cheap disposable clothing, countless new collections per year make us feel constantly out of date and encourage us to keep buying more. And that's why 80 million garments are produced each year. Okay, so 52 micro collections per year released by fast fashion brands instead of the usual 2 seasons. 400% more clothes are produced now compared to 20 years ago. 7 times average a garment worn before being thrown away. 35 kgs textile waste in generated per person per year in the US in average. But in the India, this is the positive point. It has significantly low amount to textile waste per capita. That is 0.79 kg per person. But now it's growing faster. Report says that. So only 20% to 30% of clothes of most women wardrobes are being worn. So what are we are going to see here? What are the environmental impacts of fabrics, because most of the fabrics made from the highly hazardous and harmful chemicals are using there to make the fabric. I'm sharing the examples like lycra, which is in a trend from back 7 to 6 years, and also polyester. This is completely non-biodegradable fabric. So environmental impacts, fashion industry is the second largest polluter in the world, just after oil industry, and the environmental damage is increasing as the industry grows. Environmental impacts, however, there are a solution and alternative to imitate these problems. The first step lies in building awareness and willingness to change. So here is the impact of fabric productions by chemicals, that is on water pollution too. And here is I'm explaining the water contain toxic substances such as lead, mercury, arsenic, among others. These are extremely harmful to the aquatic life and the health of millions of people living by those river banks. The contamination also reaches the sea and eventually spread around the globe. Okay, so here it is the impact on water also, and air also, and also on soil too. Also, cotton need a lot of water to grow and heat, but is dual cultivated in warm and dry areas. But up to 20,000 liters of water are needed to produce just a 1 kg of cotton. So there is a large amount of water we are using to make this. 80% of daily needs in a water used to grow cotton in a country. 100 million in India people do not have access to drinking water, says Stephen Lely from The Guardian. Okay. Uh, Now, next point is toxic clothing. So, ma'am, next slide. Every time we wash synthetic garment, polyester, nylon, etc., about 700 individual microfiber are released into water. This is also the biggest drawback when we are washing our clothes. Next slide, please, ma'am. Hello, am I audible?
Yes, Suraj, you are audible.
Hello.
Yes, yes.
Uh,
I think the
yes, ma'am.
slide is
Next slide,
lagging.
please.
Yes,
Okay, okay,
the slide is lagging, I think. Just conclude a little
okay,
faster.
okay, ma'am. ठीक है. Okay, there is. I explained different types of synthetic fabrics and also natural biodegradable fabrics in next slide. So here we need to go fast. That's why I'm continuing. Waste problems of the fashion industry also. Clothings are clearly become disposable. As a result, we generate more and more textile waste. A family in a Western world thrown away an average 30 kg of clothing each year. Only 15% recycles or donated, and the rest goes directly to the landfill or is incinerated. Okay, so here is I'm explaining. Next topic is concept is chemicals in a fashion industry. Chemicals are one of the main component in our clothes. Ma'am, next slide. Okay. Chemical industries used during fever productions, dyeing, bleaching, and wet processing of each of our garments. The heavy chemicals in a cotton farming is causing diseases and premature death among cotton farmers, along with massive freshwater and ocean water pollution and soil degradation too. Okay, here is I'm going to share one example here about toxicity of our fabrics which are making in now period. Some studies show that certain chemical substances contained in a pajamas can be found in a child's urine 5 days after wearing those pajama for a one night only. So that's why this is the biggest drawback of using chemicalized fabrics in our daily use. Greenhouse gases emission from the fashion industry, that is also the second biggest point we have to note down. Synthetic fibers, polyester, acrylic, nylon, saying etc., used in a majority, making production much more energy intensive that with natural fibers. Okay, so here is the by looking towards now condition or our lifestyle, human is not going to survive more than 100 years, except some rare examples. So that's why we do not need of the this type of fabrics which are completely entirely non biodegradable, and also after throwing it or when this come towards as a waste, they stay in our environment for 20 to 200 years still, uh, by do not biodegrading still as it is in our environment and creating many problems, screen diseases and all. Hello ma'am, next slide. Ma'am, I want to explain the other points which I am already put in my PPT. So that's why please cooperate me. Is it possible to move forward, ma'am?
Yes, yes. Can you just state which
Yes, yes,
point you
yes.
want?
Okay, ma'am.
Yes,
Okay.
yes.
Also, according to James Conga, from force, cheap synthetic fiber also emit gases like NO2, which in a 300 times more damaging than CO2. So here, by fabric soil degradation caused by the fashion industry, rainforest destructions caused by the fashion industries too, Every year, thousands of hectares of endangered and ancient forests are cut off trees used to make wood-based fabrics such as rayon, viscose, and mandol. Here is I explained different types of degradable and entirely non-biodegradable fabrics and their bad impacts on our environment too. Spandex is completely non-biodegradable. So here is clothing is defined as item worn to encloses or cover the body and does not include body modification. Clothing provides a barrier between the skin and the external environment and serves a variety of functions, which is saves protection, social and cultural purposes, and thermoregulation. Okay, impact of textiles on skin. I have already said one example about that. The chemical component seen in a urine of the baby after using that type of pajamas. So here again, next slide, please, ma'am. Here again, I explained one highly chemicalized process of making fabric. Next slide, ma'am. So at the end, what we can do and change about it. Okay, choose clothes made in a countries with stricter environmental regulation for factories, and the types of fabrics like uh, spandex and like lycra or like polyester, these are non biodegradable, so we have to recycle that and try to decrease their lifespan to stay longer. And after uh, using enzymatic biodegradation processes, we can do this. They're bleaching and dyeing again and made new products from that, and again we can use that recycled product from that. And after reusing 2 or 3 times, recycling our 2 or 3 times, their lifespan be we can decrease their lifespan to biodegrade or decompose faster. Okay, so here is some associations are doing that work, ZDHC, GOTS, and all. Government also needs to support these types of associations and also need to make a strict rules for that, to do not produce or do not use the like these types of fabrics. And also in fabric making processes, using scent and chemicals, they are also very harmful for our skin. That's why we need to concentrate on this. And here is the one example, best example. In Mumbai, Siddhivinayak temple offered flowers to Ganesh ji. They are again used as a making the colors, and they used to dye the fabrics. So we need to support these types of projects. And also here at the end, we need to use also to prevent that fungal diseases from the fabric using, we need to use herbal products. Here is my partner, Snehaji Thakre, made the complete 100% pure uh, herbal products. And here is some 100% pure products made by my partner. We give 100% surety about results. This complete our own creations of combing essentials herbs, looking towards upcoming need for the world or India. Thank you, ma'am. Thank you, everyone.
Thank you, Suraj. Again, good presentation.
Any questions?
Now, I would like to call our next participant, Ram Deshmukh. Okay, Ram. Just wait for a minute. I am going to share your PPT. Yes, Ram, you may start with your presentation. Ram?
Hello.
Yes, yes. You may start with your presentation.
Okay, ma'am. Good afternoon, everyone. Hello. My name is Ram Deshmukh. I am from TYBSc Microbiology, Department of School of Science. Today's my presentation topic is landfill composting. Or landfill. Landfill is a site. Next one. Landfill is a site for engineered disposal of waste. Disposal is by burial in the earth crust. Landfill is the oldest and most common form of waste disposal. Landfill is most common and traditional method, energy efficient process, cost effective process. Next slide, ma'am. Ma'am, next slide. Types of landfills. There are four types of landfills. First, municipal solid waste landfills uh, consist of everyday items we use and then throw away, such as product packaging, clothing, bottles, newspaper, paint, etc. Uh, Second type, industrial waste landfills. Industrial waste is generated by manufacturing or industrial process. Industrial waste include cafeteria garbage, oil, solvents, chemicals, weed, grass, etc. Next, uh, third type of landfills is hazardous waste landfills. Hazardous waste is a danger or capable of having a harmful effect on human health or the environment. They include by batteries, brake fluid, chemical strippers, chlorine, etc. For fourth type of landfills, green waste landfills. It is also known as biological waste. Is any organic waste that can be composted. They include by leaves, grass, and domestic or industrial kitchen waste. Next slide. Landfill design and operations. The planning phase is the first phase of landfill design. Uh, Planning phase, preliminary hydrogeological and geotechnical investigation as a basis for actual design. Second phase is construction phase, earthwork, road, facility construction and preparation of the fill area. Third phase is operation phase, involves work at the front of the fill area, operations of environmental installation, uh, completion of unfinished section has a high traffic intensity. Next slide, ma'am. Ma'am, next slide. Uh, Completed phase, it involves termination of actual filling to the time in when environmental installation need no longer to be operated. And final storage phase. In this phase, the landfill is integrated into the surroundings for the further purpose, no longer needs special attention. Next slide, ma'am. The process of landfill. Landfill carefully designed structure built into or on top of the ground, in which trash is isolated environment. Ma'am, screen is not visible. Okay. Hmm. The process of landfill. Landfill carefully designed structure built into or on the top of the ground, in which trash is isolated environment. This isolation creates this isolation creates a safe and controlled environment. Ma'am, next slide. Advantage of landfilling. In addition to provide an option for waste management, it also serves to improve reclaim poor quality land. Uh, it is the economical alternative for solid waste disposal, which accounts for its frequent application. Composting is a seasonal option. There are disadvantages of landfilling. Uh, Difficult to find suitable site within economically feasible distance, potential harm to public health due to air, soil, water, noise, pollution, damage to local ecosystem, public oppose. Ma'am, next slide. Conclusion. Effective design of landfill must aim at public health and safety, long-term workability of landfill, control of pest birds, etc. Ma'am, next. Hello, ma'am. Next. This is my reference, ma'am. Thank you so much.
Yes, thank you, Ram. Okay, now we are done with all the presentations and we are moving toward the valedictory program. I would like to invite Dr. Razia Putti, HOD of Department Microbiology and Life Science, School of Science, Sandip University, for giving the vote of thanks. Over to you, ma'am.
Good afternoon, all of you. It is my privilege to deliver the talk on validatory function. This was a 3-day international conference organized by the School of Science, Sandip University, Nashik, Maharashtra. And in these 3 days, we heard various eminent speakers who were either faculties or scientists or entrepreneurs, and the students from institutes and other colleges, as well as our institute, School of Science. We came across various lectures on current trends in waste management, reuse, and valorization. And we have seen bioremediation from various perspectives. The emerging contaminants were one of the major aspects which we need to recognize now and start working on the direction of removing it from the environment. We also heard lectures from very senior scientists and faculties who discussed the solid waste management practices followed conventionally, the wastewater treatment technologies which were conventionally adopted so far, and what are the various challenges now we are facing in these aspects. We came across new technologies in the area of nanotechnology, genomics, proteomics, and also valorization, how we can get valuable products from the bio waste. We heard various speakers who talked about the various bioremediation approaches, phycoremediation, then using bacterial system, using fungal system, and how we can follow up these approaches using various tools. We also heard a very interesting talk from a NERI scientist who explained how we can change this technology by adopting the digitalization. And the current era belongs to the digital world. How we can incorporate the data analysis parameters using various recent equipments, and how we can generate the data and reuse it for our own observation and management. We also came across the talk by Vermi technology. We also came across the details and the processes involved in vermi technology and vermi composting as a bioremediation approach. We also heard lectures and the various experiments carried out in system biology approach mediated bioremediation. Various young and dynamic researchers are working in all these research aspects, and they are also contributing to the publications, data generation, and guidance to the MSc and the doctoral students. The students of our college are highly benefited from all these lectures delivered during this 3 days international conference organized by School of Science, Sandip University. And I am privileged to be here expressing my thanks to all the speakers, the participants, the students, and also my colleagues who were enthusiastically involved, and we had a 3 days communication and discussion sessions on these trends and the technologies adopted by various researchers around the world. Thank you so much. Hello, Vishakha madam.
Yes, ma'am.
Yeah, we can conclude now.
Okay, yes. Now, I would like to express my special thanks to all the speakers, participants, and the audience for being here with us today. Now, we now declare the end of the conference. Thank you all. Thank you, ma'am. Now, also there is a request to all the participants to fill the feedback form. It is necessary for the generation of the e-certificates. Now, it is already being shared on the WhatsApp and the Telegram group. Thank you all. Thank you, ma'am.